So I've titled this talk, Replacing Darwin, the New Origin of Species. I'm writing a book by the same title, and hopefully by the end of this afternoon and evening, you'll see why I've titled it as such. So that Dr. Keithley has given you the stated topic. What I want to do is actually step back from this fairly specific topic and look at the really big picture of the creation-evolution debate first, and sort of a 30,000-foot view that I think is really critical in a setting like this. Then look at a 3,000-foot view. Why are we still debating a 19th century scientific topic? How many other 19th century scientific topics do we still fight about to this day? That's, I think, an important question to answer. Then spend some time, I'm calling it the 300-foot view of the non-genetic data, the non-human data, both genetic and non-genetic, and then focus very intensely on the origin of our own species. So we're going to deal mainly with the origin of species in general in this hour, and then after dinner, the origin of our own species. Now, this isn't the first time I've done a session like this. About a year and a half ago, I was at the Evangelical Theological Society meeting. I think it was held in Atlanta that year, November 2015. Spoke opposite Daryl Falk of Biologos. I got to know them some. I had spent several hours with him a couple months prior. We had about a 4-hour lunch. Interact with Jim Stump, with Brad Kramer. I had dinner. We had a lovely time. He's their managing editor. And he said something to me at the end of that dinner that I thought was really insightful. He says, and he has an MDiv degree. He said, look, I'm not a scientist. And for all of us who are not scientists, we can't adjudicate the evidence. We're just not trained. So it really comes down to a question of who you trust. And I thought that's very true. If roles were reversed, if this was, let's say, a discussion of Hebrew, and some of the Southeastern Old Testament faculty were discussing, I'd be sitting in the audience having to make that same decision. I can't adjudicate ancient Hebrew. If it was a physics discussion, I can't adjudicate physics, geology. It'd be a question of who I trust. So that's, I think, the bigger question everyone has to answer. Anyone who's not necessarily a biologist or geneticist, that's the question you have to answer by the end of this evening. Now, that question becomes all the more acute once you look at the statistics, which the Pew Forum has been supplying to us for a number of years. There we go. And the surveys have shown a fairly consistent view of creation among the general populace for many years. Here's what I'm talking about, though, in particular. The Pew Forum has done surveys of the American Association of the Advancement of Science members, or AAAS members, and the results are very stark. If you look at the number of AAAS members who would say, evolution is the explanation for how humans got here, and we got here by natural processes. 87% of professional scientists would say this. If you include those who say God guided evolution, you're up to 97%. In other words, less than 3% of professional scientists would endorse what I'm going to tell you this evening. So who do you trust? And really, though I'm here on behalf of Answers in Genesis and Dr. Venema on behalf of Biologos, we're really representing this bigger, wider cultural debate. And so, as you think about who you trust, those are the numbers you have to deal with. And I'd like to take it a step further and say, whichever position you pick, you then have to explain the other side. So let's say, you say, well, how can 97% of scientists be wrong? You're then going to have to explain the 3%. And there's a number of plausible explanations you could think of to explain why dissenters exist. Especially in the general public, you might say, well, they're just not educated on evolution. They haven't learned the science. Maybe for those who have been trained in the science, they have such a strong religious commitment to a particular biblical interpretation. They speak outside of their field. And so, this can't be true, but they're really not trained in that. So those are all potential explanations. But I think there are some particular individuals, like my colleague Jeff Tompkins, who worked at the Institute for Creation Research. I worked with him for 6 years there before coming to Answers in Genesis. He was faculty at Clemson University before coming to ICR. He came to ICR around 2008, 2009. 10 years at Clemson, 5 years as director of the Genomics Institute, has over 50 published papers in the field of genomics dealing with all sorts of organisms, animals, plants, bumblebees, cotton, so forth. He's done an enormous amount of work on this topic. And his area of dissent is in the field of genomics. Some of his intense focus has been on the human-chimpanzee genetic similarity, the idea of the human of a chromosome fusion, and human chromosome 2 and our descent from the chimpanzees, according to evolution. and other such arenas. So he's speaking in the area in which he's done professional work. So if you trust the 97%, what do you do with people like that? And of course, the question goes both ways. So that's, I think, the explanatory task before us this afternoon and evening. And I think in some case, so what I'm going to do now is I'm going first, so I'm going to deal with what's been published, and then hopefully by the Q&A we can interact more directly with one another. So what follows is not to be disputatious, it's just because I'm going first. So, how would some of the 97% explain people like me? So again, I'm just taking from Dr. Venema's book because we haven't sat and discussed this ahead of time. Just going on what he's printed. So he said, it's no exaggeration to say that the very, very few trained biologists, less than 3%, who reject common ancestry do so because of prior religious commitments, not for scientific reasons. And my impression is that's probably what most of the 97% would say. The Pew Forum has also surveyed religious profession among professional scientists, and at least 2/3, and I can say this now in a seminary setting, would not be born again. Why is that? Because at least 2/3 cannot positively affirm belief in God, and belief in God is a necessary but not sufficient criteria for being born again. So at least 2/3 are not Christians. And so it wouldn't surprise me if many of them would say, well, people like me who have a strong religious background, well, it's his religious commitments that are pushing him to embrace this particular view. And then to support this, Dr. Venema has quoted Todd Wood. Todd Wood would call himself a young earth creationist. Now, the term young earth creationist is not copyrighted, so anyone can take it who wants to. And you can probably find a young earth quote for anything you want, including things like cancer is not caused by mutations. There's also all sorts of crazy ideas out there, all sorts of splinter groups, and what goes along with that is also lots of lack of accountability. And some of what we do at Answers in Genesis to correct that is we have an internal editorial review board, we seek outside peer review. There's all sorts of ways to try to combat that trend in the young earth community, which is to be your own camp, have a group of followers, and have no accountability. Anyway, Todd Wood would fall in that splinter group category. So he says evolution is not a theory in crisis. There's evidence for evolution, gobs and gobs of it. It's my own faith choice to reject evolution. Now, Todd was pressed to give evidence, and then he said in his blog he wouldn't. Anyway, that's an example of a typical explanation, let's say, for the 3%. But I think Dr. Venema has taken another step further, so this is something I want us to consider. He says Todd Wood is just being honest about the state of the evidence for evolution. But what does that imply then about those who would dissent? Would that imply that people who would disagree with the evidence for evolution are being dishonest? It's a question to consider. In one of the footnotes, Dr. Venema says Todd's approach is unique among the young earth literature in that it is thoroughly accurate and does not misrepresent the data. Does that then mean that the rest of the young earth literature, Jeff Tompkins and others, is inaccurate? Does it misrepresent the data? Now, that's from Dr. Venema's book, because that was the stated topic for the reason for the invitation to discuss both of our books. I want to broaden the view now and ask more generally what do the 97% view. So that same 2015 ETS meeting, Biologos blogged about, and so this screenshot here is from their write-up from that 2015 event. And like their blog post, they have comments. One of the commenters, and so this comment is not a Biologos staff member. Anyone can comment. But you'll see why in a minute why I'm citing this. It's a gentleman named Patrick, not a Biologos guy. If you can't see that, I'll read it for you. He said, Jeanson, meaning me, may appear to be quite articulate. He is still dishonest in saying he is doing real scientific investigation. He reads the same genetic papers that I read. Granted, he's more knowledgeable in genetics than I, as he has recent degrees in the subject and has published papers in the field. However, he takes the results of researchers who painstakingly worked years squeezing out new scientific knowledge from new genetic sequencing technology and dishonestly manipulates the data to correspond to Ken Ham's view of what the Bible says. That is not ethical scientific research. It is not science. He gets paid to do this dishonesty. He purposely manipulates other scientists' research to fit his and Ken Ham's views. It is dishonest and borders on fraud, can be harmful to society and individuals if anyone takes him seriously. Now again, this is not a spokesperson for Biologos, and this is not what Biologos has said, but I guess my question is, there's 60 plus comments on this blog post. Why was this not flagged as inappropriate or as character assassination? And I talked to Jim Stump after that 2015 ETS event, and he also floated the word dishonest in our discussions. I asked him for his evaluation. Now, Biologos' core commitment is to strive for humility and gracious dialogue with those who hold other views. So I found this a little puzzling, something we can discuss further. Now, I'd say consistent with this pattern, though, I want to cite one more example from Joel Duff. So Joel Duff is a member of the Biologos Voices, though he has his own blog and publishes frequently and has many criticisms of the young Earth model. He wrote this article for Biologos in August 2016 in response to the opening of Answers in Genesis Ark Encounter. And you can see the topic of his several hundred-word blog post, Did Modern Animals Evolve from the Inhabitants of the Ark? I'll explain what he's getting at in a moment. I would describe this from my perspective as a hit piece, and you don't have to agree with this. Why would I say that? Well, let me just give an example. So he says, is there evidence for the rapid post-flood speciation proposed by AIG? Let me explain those terms. Dr. Duff does here. He says, if there were only 1,400 or fewer kinds on the Ark as claimed on the Ark Encounter, and that's accurate, that's what we would say, that kinds, so from a young Earth perspective, kinds and species are not the same thing. If you think of the modern taxonomic system, you have species, next level up is genus, species, Homo sapiens, that's our classification as humans. The next level is family, order, and so forth. We'd probably not put it at species or genus, but generally, at least for vertebrate species, more at the level of family. As an example, there's 37 or so living cat species. We'd say they're all part of the same family. We'd say they go back to 2 ancestors on board the Ark. So what Duff says here is correct. If there are only 1,400 or fewer kinds on the Ark, as claimed on the Ark Encounter, and today there are more than 30,000 land animal species, let me stop there and explain that as well. So we'd say Noah took 2 of each kind of land air-breathing species, so that's just a handful, as he said, a few thousand. And from those then have descended the tens of thousands of land air-breathing species we see today. So if that's true, he says, and countless extinct species, is there evidence for the sudden appearance of thousands of new species in just over hundreds of years? Actually, it's over several thousand years, but whether hundreds or thousands, it's obviously in a stark contrast to what the evolutionary model would say, which is over millions of years. So how do you get this species sprint? That's his question. It's a fair question. Is there evidence for it? That's the question he asked. His answer is absolutely none. That's just the tone he takes in the article. Now, what's puzzling about this is I personally have written over 90,000 words on this topic alone, several technical papers, a lay article series, all that was published before his article was posted. Now, he asked another question in this article, where do young Earth creationists seek evidence for rapid post-flood speciation? You'll notice there's a placard there that's a picture of a placard on board the Ark. It seems that he tries to answer that question largely with the placards and lay-level articles, because when you look at the references, you don't see any reference to any of the technical papers or more scholarly work that young Earthers have published on this topic. Now, if you believe your opponents are liars, that's consistent, and that's well within anyone's right, but it was puzzling. Now, I'm interested in dialogue. I think this is very productive. I actively seek out people, especially opponents, who can critique what I'm doing, both for reasons of integrity and for reasons of pure selfishness. Who wants to publish something that's wrong and then get reviewed and be wrong? And you get publicly exposed. So I personally like to know privately ahead of time. Anyway, so I wrote a response to what Dr. Duff had written. He only wrote a couple hundred words. It was a lay-level article, and I was trying to give an answer on some of his technical points, so mine was like 9,000 words, just because it takes that much time to then say, is this what he said? Explore it. What are the answers? Here's some data. And about a month ago, Dr. Duff wrote another article for Biologos in response to the new movie, Is Genesis History? This was not necessarily on the topic of speciation, but that topic came up in his article. Get my slides synced here. There we go. That was his recent article in March. Though he referenced in this article this rapid speciation concept. He talked about, there we go, the impossibly fast pace of speciation. That's hyperlinked. It hyperlinks back to the August article with no comment on my response, as if it doesn't exist. Now, at the ETS meeting, Daryl Falk and I were speaking, this is, again, a year and a half ago. Daryl Falk and I spoke opposite of one another on a similar, more narrow topic, just the Adam and Eve question, not broadly creation, evolution, genetics. And we had, like I said, we met for 4 hours a couple months prior, had a lovely time. And he said, it's a shame your organization won't allow more of this sorts of dialogue to happen. I said, I'm not sure that's true. I can't speak for the leadership. I said, where I could really use some feedback and some dialogue is on my technical papers. He said, well, I'm retired now. I don't have time for this. I don't think I can get to it. At the ETS meeting, and you can get the audio recording of this, he also brought up the topic saying, it's really too bad we don't have more dialogues. We need to have more discussion. And I said, well, maybe something's changed. I said, Dr. Falk, we talked in September about you potentially reviewing my papers. Has it changed, basically? He said, no. I said, maybe. Now, we haven't discussed it since then, so the offer is still standing. So that's just a sketch then on this broader question, who are you going to trust? And how do you explain the other side? I want to set in front of you now, so I've just set in front of you what the 97% might explain the dissenters. Now I want to focus briefly on theology. I'll leave my theological scholarly friends to discuss the theology. Just one point I want to make here. So the Biologos statement of faith speaks of the Bible being inspired and authoritative. You will not find a positive affirmation of inerrancy. And Deb Harzma, the Biologos president, has told us why. This is also from one of the Biologos articles. Some in Biologos would not be comfortable with the word inerrancy. It's not how they would characterize their view of Scripture. Now, it would seem to me that Biologos, and the 97% in general, would seem to take a very serious view of the scientific consensus. And I'll give you some more quotes in a moment towards that end. And so much so that it seems like the dissenters would be labeled liars if they disagree. Why is it then that a similar commitment is not made to the Scriptures? That's a question I think we need to discuss. Now, again, my focus is in general on the 97%. I'm starting with Biologos because that's obviously of immediate relevance, but now I want to go broader. Let's go to the American Scientific Affiliation, a 2,000-member organization of professing Christians as a statement of faith. How would they characterize the dissenters? So Randy Isaac was president for about 10 years, formerly a member of the Biologos Advisory Council. He was interviewed, sort of an exit interview, I think about last fall, in their magazine, their journal, Perspectives on Science and Christian Faith. And this is what he said about, let's say, a young Earth creationist view. He said, claims that scientific data affirm a young Earth do not meet the criterion of integrity in science. The ASA can and does oppose such deception. So my impression is that this is a more broadly true, an explanation for the 3%. So whichever side you pick by the end of this evening, 97%, 3%, or maybe make up your own, to me the explanatory task is what do you do with the other side? If you pick the 3%, what do you do with the 97%? If you pick the 97%, what do you do with the 3%? That's just a 30,000-foot view of this debate. That's, I think, the big picture, though, I think especially relevant to anyone who's not a geneticist. Like I said, it would apply to me if physics was the topic and not genetics and biology. Now I want to try to answer the question, why are we still having this debate? Why aren't we debating spontaneous generation? Why aren't we debating some other scientific view from the 19th century? I think the answer to that question is found, and we can find it just upon a brief reflection on the topic that Darwin wrote. So now I'm zooming in from the big, broad question to now biology specifically. Darwin's topic was biology. His book was titled On the Origin of Species. Why are we still debating the origin of species? Obviously, most of the scientific community is not, 97% are not, but why is there still remnants of this to this day? Well, what is a species? That's the question. And that's the answer to that question, I think, holds the answer to why there's still a debate. We intuitively recognize what a species is. We recognize zebras by their stripes. There's actually 3 different species of zebras that differ in their stripe patterns. This is the plain zebra. It has stripes that go all the way around its belly. The other 2 do not. They stop. We recognize species by their traits. Zebras by their stripes, elephants by their trunks. And there's 2 or 3 species, depending who you talk to. You can recognize this African elephant species because of its shape, the shape of its ears and overall anatomy and physiology. We recognize different cat species, large and small, tigers by their stripes, and what I've tried to show in each of these images is offspring. This is a kindergarten point, but it's really profound. Species produce like individuals. If a tiger could produce an elephant, which could produce a snake, which could produce a butterfly in one generation, or one generation after the other, we wouldn't even have the concept of species. The fact that we have this term in our vocabulary implies some sort of consistency. Not necessarily perfect consistency, but it implies some sort of consistency. So let's think about this then. Species are defined by their traits, and traits are inherited consistently generation to generation, or at least the instructions for those traits. We now know those traits are encoded by genetics. So species are defined by traits, traits are defined by genetics. If you want to know the origin of species, if you want to know the origin of this tiger species, you need to know the origin of stripes. If you want to know the origin of stripes, you've got to know the origin of the DNA sequence that encodes it. If you want to know the origin of species, you must ultimately know genetics. The origin of species is a fundamentally genetic question. Now, how much did Darwin know about genetics in 1859? He tells us. This is from the summary section of chapter 5. He says, our ignorance of the laws of variation is profound. Well, he doesn't use the term genetics. That's because the term wasn't invented until the early 1900s. Most genetics textbooks begin with Gregor Mendel, and his results were published in 1865, 1866, 6 years after Darwin published the first edition of his work. Mendel's results were lost for 30 years, rediscovered around the turn of the century, connected to chromosomes. Chromosomes are made up of protein and DNA. It took another half century, basically, before the scientific community accepted DNA as the physical substance of heredity. So nearly 100 years after Darwin published his work, the scientific community finally decides DNA is where it's at. And of course, like every good scientific story, that ends one and begins another. If you know that DNA is the substance of heredity, then the question becomes, what is the DNA sequence of the species around the globe, and how did it arise? So 1859, we don't have any DNA sequences. 1953, structure of DNA, Watson, Crick, and colleagues. Late 60s, we have the first DNA sequences that come into the databases. Late 70s, the first genomes. You start small. The viruses were the first species that had their complete DNA sequenced. 1997, which is what I'm demarcating there, is when we passed the 1 million DNA sequence mark. That doesn't mean we had the DNA sequence from a million species. I bring it up as significant because there's 1.6 documented species alive today, with conservatively many more left to be documented. My point is, in blue there, that's when we can finally answer the origin of species directly for the very first time. In other words, Darwin took a gigantic scientific risk when he tried to answer a fundamentally genetic question nearly a century and a half before he'd have any genetic data with which to answer it. And I think Dr. Venema would agree with me based on, or the substance of what I've said based on what he wrote in his book. That, of course, raises the question. If he took this massive scientific risk, how then was he able to persuade his colleagues? Why are his views still so persuasive today if we're at really the beginning of the modern genetic era? So that brings us then to the third topic I want to cover, which is a 300-foot view of genetic and non-genetic data in species other than humans. Now, the first chapter of Dr. Venema's book has an excellent description of how science works and how theories arise, and I want to just pull some quotes from it so we're all on the same page. So he says, the theory is an explanatory framework for why the facts are the way they are, and connects it to his personal story in a compelling way. In science, a hypothesis that is not rejected after many, many predictions and tests eventually becomes a broadly explanatory framework that has withstood repeated experimentation and that makes accurate predictions about the natural world. In other words, a theory. That's a pretty big quote, and we're going to focus on different parts of this, both this hour and the next. I want you to focus right now on what I've highlighted here. A hypothesis that is not rejected becomes a theory. That's the key to understanding how Darwin could persuade his colleagues in 1859, long before anyone would have any genetic data. Dr. Venema, of course, would say that evolution is a theory in the scientific sense because it's withstood 150 years of scientific testing that has failed to reject it. So let's explore some of that. I'll begin with the non-genetic data, sort of chronologically then, and then get to some genetic data, again focusing on non-human species. We'll focus on humans specifically after dinner. So what are some of the things Darwin might have cited to argue for his ideas that would have persuaded his colleagues? We have to put this in a historical context. What's going on in 1859? What do people believe then? Let me back up even further. The origin of species debate is really recent in a broad view. What are species? We just discussed that. Well, species also denotes a formal scientific term of classification. That goes back to Linnaeus in the 1700s. Before Linnaeus, by and large, there was no universally accepted term for species. Therefore, if you don't have the term in your vocabulary, you really can't have a debate about it. Linnaeus, the father of modern classification, writes his Systema Naturae in the 1700s, the 10th edition, 1758. If he's the father of modern taxonomy, you wouldn't expect the 10th edition to have that much documented, and if you look at the numbers, that's true. Let me use an example. So we're classified as mammals. There's over 5,400 mammal species alive today. How many do you think Linnaeus had documented in 1758? Less than 200. Now, if you're in the 1700s, early 1800s, and you have a very small data set of what actually exists on this planet, think about it. When was Australia discovered? What was missing from Linnaeus' data set? A whole bunch of marsupials. If you don't have a good sense for what species are alive, where they live, and really what the world is like, you might reach conclusions that are, in retrospect, fairly premature. So one of the conclusions that Linnaeus and his contemporaries reached was the idea that they looked at how certain species fit their environments. You might go to the Arctic, or the polar bear fits its environment very well. Seems designed for the Arctic environment. You go south, North America, black bears exist, not polar bears. They fit the North American forest environment better. So without even considering the scriptures, you can see how some of those early scientists derived a view of species fixity. Well, you can see where this is going. It wouldn't take much then, much scientific advance, much more discovery of species to reject that view. So that's largely what Darwin was rejecting. What he did is he corralled multiple lines of evidence. We don't have time to go into the geographic arguments he made. You can make an argument for common ancestry from geography alone. What he showed was that these data rejected the hypotheses of species fixity and the fixity of their geography. So he argued for migration and common ancestry. And he showed that these data did not reject evolution. That's how it works. That's how science works. Now, if the pendulum is here, no species are related. They all have been in their present locations since the beginning of time. Darwin swung the pendulum completely over here and said, all species are related and they have acquired their modern locations by migration. Now, what are some of the data he would use to argue for that? What are some of the data he would say failed to reject the hypothesis of universal common ancestry? One is the pattern of life, the pattern in which life is classified. Technically, we'd call it a nested hierarchical pattern. It's a groups within groups, kind of like Russian nesting dolls. So humans, Homo sapiens, Homo genus species sapiens, are classified as a single species because we all share a tremendous number of characteristics in common. Wherever you go around the globe, tremendous number of characteristics. So we form a single species. Now, we have more in common with the great apes than we do with any other species alive on this planet. We have less in common with great apes than we do with one another, but more in common with great apes than anything else. So we're classified today in the same family, Hominidae, with the great apes. Now we have more in common with monkeys and other primates than we do with other species. Obviously less in common with monkeys than we do with one another, but then we form the same order, primates. That's how this principle works. Groups within groups pattern. As you ascend this hierarchy, species, genus, family, order, class, phylum, kingdom, the number of shared characteristics declines, but the number of members of that group increases, and then the reverse pattern holds true. Kingdom, Animalia, all animals have a certain number of characteristics in common that we share, that we do not share with plants. And then you go down the hierarchy, the number of shared characteristics increases. We have more in common with mammals than we do with the rest of life, but the number of individuals in the group decreases. We have more in common with vertebrates than invertebrates. We have more in common with animals than we do with plants. That's the pattern, the nested hierarchical pattern of life. Why would evolutionists say this fails to reject the hypothesis of common ancestry? Because it fits what Darwin proposed as the mechanism for the origin of species, descent with modification. So you can see the first split there is between invertebrates and invertebrates, so they would have split millions of years ago. And once their lineage is split, they're acquiring differences, their modification, the modification element of descent with modification. So the number of shared characteristics is sort of a function of time. And so they'd say as life evolved and diversified, the number of shared characteristics is a function of time since common ancestry. So the groups within groups pattern fails to reject the hypothesis of universal common ancestry. Now, what would creationists do today? Why they're still dissenters then? Because the modern creationists are not species fixity, and I'm speaking now for the young earth creationist community. Like I said earlier, in talking about Joel Duff's article, we'd say God created kinds, mean in Hebrew. Maybe I'm butchering that, you Old Testament people can tell me, but that's my best attempt. And that it's not equivalent to species or genus, and we can talk about that later as how we derive that. In short, it deals with breeding tests and so forth. But really scientifically, it comes down to genetics. Anyway, it's a whole other separate topic we can discuss at length. The point is, modern creationists do not share the same views as the 1859 creationists. We'll get to the implications of that a little bit more later. One of the things that also has changed since 1859, you have the intelligent design community that has done a lot of work in developing the design side of the creationist model. What we'd expect from a design perspective. If God designed life, fiat, instantaneously, what would we expect him to do? Now, Genesis 1 does not tell us that God used this engineering textbook and design principle and so forth to make creatures. But what we do have is the statement that God made man in his own image. I know theologians still debate what this means. I think we'd probably agree that at a minimum it means that we somehow reflect something about the divine. So then I think it's fair to say, let's look at how humans design things and the patterns in which they design things, and see if that can inform the question of how God might have designed things since we're made in his image. So I'm going to use vehicles as an example. You can look at sedans, 4 doors, 2 doors. There's a certain number of characteristics. Skip that slide because I'm out of order here. Here we go. Sedans have a certain number of characteristics that they all have in common, and there's a whole number of characteristics they have in common that they do not share with other vehicles. Now, if you expand your system of classification of vehicles to those with 4 wheels, sedans and trucks will have a number of characteristics they share that they do not share with 18-wheelers or tractors. But sedans and trucks have less in common than sedans in general have in common. And you see where I'm going with this. Land-based vehicles that are powered, so 18-wheelers, pickup trucks, sedans, you name it, Indy cars, all have a number of characteristics in common that they do not have in common with jet airliners. But all powered vehicles have something in common, namely an engine, that unpowered vehicles do not. My point is, the things that humans have designed naturally fall into nested hierarchies as well, and the parallels are really astounding. What I'm not saying is that there are first principles that require this. You could design vehicles any way you wanted to. But it just so happens that these fall out into a nested hierarchical pattern. Now you might say, well, sedans themselves don't form a nested hierarchy, whereas species do. And I might say, well, panda bears don't form a nested hierarchy. They're a single species. So there's parallels even there. If you're comparing equivalent levels of classification, the parallel is really striking. So I would say that the nested hierarchical pattern of life also fails to reject the hypothesis of design. Fails to reject the hypothesis of universal common ancestry, but now, 150 years after Darwin, I would say it also fails to reject the hypothesis of design. What does that mean? Well, we'll get to a few more evidences and then reflect. Let's use another common evolutionary evidence, homology. It's similar but slightly different from the argument I just presented to you. Homo and homology, the study of homo, the study of sameness, or the study of biological similarity. If you were to strip away the muscles, the skin, the nerves, and so forth from your forearm, you'd strip it down to the bone. It would look kind of like this on your left. It's color-coded here, so you can see the order of the bones from top to bottom, and you can see there in brown, 1 bone at the top, then in red and white, 2 bones, then in yellow, little blobs of bones, and the fingers. And you see that same order of the bones in the dog forelimb, the bird, and the whale. Now, there's obviously differences at the finger level, and evolution is descent with modification. The differences are due to modification. Evolutionists would say, why is there this shared structure, shared order of the bones? Because they were inherited, descent with modification from a common ancestor. So this evidence fails to reject the hypothesis of universal common ancestry. Well, in 2017, does it reject the creationist hypothesis? And once again, the design community has done a lot of work on developing what does a design model predict. And I would then apply to Scripture and say humans created in the image of God. We reflect something about the divine. How have we designed things? Well, you look at, again, sedans. There are things they have in common. There's a shared anatomy. You can see these 3 here on the screen. A Volkswagen product, Ford product, Mazda. So their manufacturers are based 3 different continents. Why do they have headlights in the front, 4 doors, hatchback, 4 wheels? Why do they have doors on the side? Is it because they inherited these features from a common ancestor in the Arctic? Obviously not. Is this the only way to design sedans? No. It just so happens that they have these shared features because it's easier to get into the car from the side than through the roof. It's a basic empirical observation. So I would say homology fails to reject universal common ancestry. It fails to reject design. Well, what about transitional forms? Under the evolutionary model, if we started in the sea at some point, then fish-like creatures would have to evolve onto the land and go through a series of intermediates. And evolutionists would say we found some of those intermediates, one of the most prominent being Tiktaalik, or the fishapod. Seems to have scales like a fish or features like a fish, head like a tetrapod or land creature, and fins that seem like limbs that seem half like fins, half like legs. Seems to blend the features of two very different creatures. Would seem to fail to reject the hypothesis of universal common ancestry. So you can probably guess where I'm going with this now. Does it reject the hypothesis of design? Let's switch from commercial vehicles to military. What do military engineers do? How do they design things? If they want to move firepower through the ocean, if you've watched the news in Syria lately, you might have seen something like this, firing Tomahawk missiles. They design very powerful means of moving missiles and other firepower through the waters. Now this ship is pretty useless on land. It's a sitting duck. It'll get blown to smithereens. If military engineers want to move firepower on the land, they design all-terrain vehicles or tanks. These will sink like a rock in the ocean, are useless there. That's why they build ships. Now, what if you want to move firepower from the ocean to the land or vice versa? Military engineers have designed transitional forms, amphibious assault vehicles, that blend the features of ships and tanks. They aren't as good as ships in the ocean. They'll move through the water. They aren't as good as tanks on the land, but they'll do the job. They are perfect for the transitional environment between water and land. So I would say the existence of transitional forms also fails to reject the hypothesis of design. So where does that leave us? By this point, you might say, are there any evidences that do reject the hypothesis of design? If Darwin could reject fixity of species and the fixity of their geography in 1859, what's left? Is there a way to reject this design evidence? And ever since Darwin, evolutionists have been saying yes. And here's an example that combines this idea of ancestry and design. You can find it, I think it's in Jerry Coyne's book, Why Evolution's True. Well, this is from a Nature paper. A diagram of, so this is, I think, a couple decades old, but we'll get to the point in a moment. Diagram of the evolutionary series of fossils and I think some modern species here, showing the origin of whales. Evolutionists would not say that whales originated directly from a fish-like creature. They'd say that whales arose from a land mammal, four-legged land mammal. Well, as that evolved back into the ocean, no longer any need for those hind limbs, so that they've basically degenerated into this stump, this remnant, this vestige of a hind limb. So what purpose would this serve? Why would a designer put the stamp of an evolutionary history in a creature like this? Why does this exist? It would seem to refute the idea, seem to reject the hypothesis of design while simultaneously failing to reject and perhaps even confirming the hypothesis of common ancestry. So what's the answer from a design perspective? Well, it's true, these don't seem to serve much function in the purpose of locomotion. But whales exist today not just for the reason that they move through the water, they exist today because they reproduce. And reproduction in the water, the buoyant and slippery environment of the water, is a little bit more challenging than on land. And without getting into too much of the birds and the bees of whale reproduction, a little bony help at the back can make it easier. And that's all I want to say about whale reproduction. If you look at our own body, you can find evolutionary arguments there. In fact, when I was in Texas with ICR for 6 years, the Perot family funded a new science museum. It was originally going to be subtitled Exploring the Wonders of Evolution. They have, I think, on the second floor or third floor, a whole section dedicated to these sorts of arguments, but within the human body. And a classic example of this has been the existence of the human appendix. Seems to serve no function. In fact, it was ridiculed for many years as existing only to line the pockets of the surgeons who were paid to remove it. So what purpose does it serve? Well, even Jerry Coyne now would concede it serves to function in the immune system. He would argue that the size of the appendix fits the evolutionary idea, that it fits diet. Well, that's been rejected as well. Anyway, the appendix has a function. What about in our skeletal system? Look at our spine. What's at the end of our spine? The tailbone. Evolutionists would say this would seem to hearken to our evolutionary past, our monkey past, prehensile tail. Once we moved out of the trees, devolved out of the trees, no longer any need for it, and so it's degenerated to this stump, tailbone. Doesn't seem to serve any function. Well, if you know someone who's had their coccyx, that's the term for it, the coccyx removed, And if you know them very well, you can ask them how their bowel movements are doing, because the coccyx is the site of attachment for many critical pelvic floor muscles. And I'll spare you the details of that as well. Needless to say, it has a function. And there are many such other examples like this. Now, the topic, of course, for this hour is creation, evolution, and genetics. How has genetics changed this? And from my impression is, and we can discuss this more again next hour when it comes to humans, my impression is that genetics has changed this very little. If you look at evolutionary textbooks, they're still making the same arguments that I've just shown you, but with DNA. They'll say if you look at the DNA of humans, it fits a nested hierarchical pattern. And I'm not keeping up with my slides because I'm looking at the podium. My apologies. There we go. If you look at the human DNA, it falls into a nested hierarchical pattern with primates, mammals, vertebrates, so forth. This, again, they would say, fits this idea, fails to reject the hypothesis of descent with modification. And because we've already covered this, I'll just say I would argue it also fails to reject the hypothesis of design. If the final structures are the same, the blueprints are going to be shared. Why am I saying blueprints? DNA is really the blueprint of our body. It's the instruction manual for 10 fingers, 10 toes, a nose, 2 eyes, so forth. Well, if the sedan's final structure falls in a nested hierarchical pattern with other vehicles, well, then the blueprints for these vehicles surely will as well. So I think the same type of argumentation holds here. You can even find a DNA argument that fits this idea of homology. Rather than saying there's shared structures, evolutionists would say there's shared genes. So genes are just subsets of our DNA that encode a particular function. They might say, well, we know the genes, in this case they're the Hox genes, called something else in fruit flies. We have a similar set of genes as fruit flies that specify our body pattern from head to trunk. Why do we use the same types of genes? Fruit flies, mice, humans, why is that? Descent with modification. Descent because we inherited them. They'd even say, the structure of chromosomes, the patterns and the linear arrangement of these genes on chromosomes is shared. The reason that these are shared is descent with modification. Once again, though, we're looking at the blueprints. If the final vehicles have shared anatomy and so forth, structure, well, then their blueprints will have shared things as well. The blueprints will have things in common. Again, not due to common ancestry in the Arctic, but simply from design principles. Not from first principles, let's say. This is the only way you can design it. This is simply an empirical observation. Because the DNA sequences that we possess are largely from living species, we don't yet have a very good analogy for the transitional forms argument. Maybe you could argue with some of the limited data we have. So I'm not going to cover that, but the parallels, again, to the anatomical arguments find an analogy in DNA. Is there genetic evidence that rejects the hypothesis of design? If I'm using design as a competing explanation for the data we just looked at, what about at the genetic level? And of course, Biologos would cite this. If you look at their genetic evidence, they talk about genetic scars, which, again, by its name, implies some sort of error. And I'm not trying to misrepresent them. You'll see some of the detailed arguments are arguments from function. So what is the function of DNA? How much of the human DNA is functional? Some evolutionists might say, well, maybe 10% is functional. That's all you can argue for. Let's think back to the evolutionary arguments for the appendix, for the tailbone or the coccyx, for the evolutionary vestige. What was the problem with those arguments? I'd say there were two problems with those arguments. Number one, they were arguments from silence. Absence of evidence is not necessarily evidence of absence. I'll say it again. Absence of evidence is not necessarily evidence of absence. It might be, but it might not be. So it's already on a shaky foundation. What makes it even more shaky, and I think a second reason these arguments faced some severe challenges, is the absence of evidence was not derived from decades of experimentation that failed to identify positive evidence for function. The absence of evidence arose from the absence of experimentation itself. So then no surprise, as experiments progress, that evidence of absence disappears and we find evidence for function. So when evolutionists might say there's little evidence for function in the human genome, the first question to ask, of course, is, well, what experiments have been done? The gold standard is genetic knockout. Fortunately, that's still considered unethical in humans. We shouldn't be guinea pigs where someone goes into you and tries to knock out your DNA just for knowledge's sake. But there are individuals around the planet that have naturally because of mutations, because I would say because of the fall, they're missing sections of DNA, or somehow some of their genes have been rendered non-functional. Now, only 6%, I think this is an Icelandic study, only 6% of the genes in the human genome have this sort of individual representation, which means the vast majority of our genes have yet to be tested by the gold standard for function. And since genes represent less than 5% of our total DNA, you can easily see then that the vast majority of our DNA sequence has yet to undergo any experimental manipulation. There's been biochemical studies, you may have heard of the ENCODE project, that they said there was evidence for function in 80% of the genome, and it was criticized as being premature. It is premature. It's only biochemistry. It's not genetics, and they're only looking at a small subset of cell types. There's all sorts of developmental windows they did not have access to, and again, you're running into ethical issues here. What are you going to do? How are you going to access a baby in the womb and test the function of DNA in those tissues? But I think we have the same problem as we do in anatomy and physiology. Absence of evidence is not necessarily evidence of absence, and the absence of evidence is from absence of experimentation. To me, what the ENCODE project suggests is a trajectory that is pointing towards increasing levels of function. We won't know if that trajectory holds up or not, but it looks to me like the evidence is pointing towards high levels of function in human DNA. And again, that's what it looks like to me. That's a hypothesis that's testable. And I anticipate then that as that level, the evidence for function goes up, the arguments then will have to necessarily change. And let me just pick one example real fast for sake of time. This is the ENCODE project. I'll skip that again. There's a study of pseudogenes. So you can see in the name pseudogene, even in the name itself, it sounds like this was a gene that once was and no longer is. It used to be a gene. It's broken. Well, the ENCODE project studied basically every single letter in the human DNA sequence. And since there's pseudogenes in the human DNA sequence, it's no surprise these were studied as well. Well, similar experiments have been done in other species, including worms and fruit flies. And the pseudogenes in these species have also been investigated biochemically for function. And not surprisingly, the number of human pseudogenes that have at least preliminary biochemical evidence for function mirrors what you see across the entire genome, about 80%. And similar numbers, 70-ish% in worms and flies, also hold true. Again, this is not genetic knockout experiments. It's not the gold standard. But it does set a trajectory. I don't think the evolutionists anticipated this sort of thing. Only time will tell how this turns out, but again, in light of what anatomy and physiology has shown, I don't think this trajectory bodes well. So let's wrap this up then. Where does that leave us? We went from Linnaeus in 1758 to the present, with Darwin in between. Darwin rejecting, through a series of very clever experiments, very clever data, was able to reject the hypotheses of fixity of species, fixity of their geography. Also argued that there was a number of lines of evidence that failed to reject the hypothesis of universal common ancestry. And this is why Dr. Venema would say that 150 years later it still stands. I would argue, though, looking at the classic lines of evidence from evolution, both in the realms of anatomy and physiology and genetics, and tracing the origin of modern young earth creationism, or at least the revival of young earth modern creationism, to the publication of the Genesis Flood by Whitcomb and Morris in 61, about 50-ish or so years ago, that the young earth model has also withstood years of experimentation and tests. So there's a number of lines of evidence that failed to reject the young earth model. Now, part of Dr. Venema's definition of a theory is one that makes testable predictions. That's something we'll explore in detail after dinner. But on the perspective in general on the origin of species, I would argue that what we see here is a rejection of one end of the spectrum by Darwin's data, species fixity and the fixity of their geography. Darwin swung the pendulum the other way with a bunch of lines of evidence, and I would argue that those lines of evidence do not reject the modern creationist view, which basically means we're back to square one in the origins debate, which means then what we're going to discuss after dinner is going to be doubly important. Number one, because it's genetic data, and it's our own species, so there's a human interest factor, but it's genetic data that we're going to focus on intensely. And it's the most important scientific field. Genetics is the only direct record of a species' ancestry, scientifically, and the only way you can use indirect data is by rejecting competing views. And hopefully what you've seen is those competing views have matured and makes the debate different. And in light of that fact, then, it's doubly important that we discuss this genetic data in depth in the next hour.
I'll just say again, it's a delight to be here. Thank you so much for the invitation to Dr. Keithley and Southeastern to have me here. It's been a delight to get to know my brother in Christ, Nathaniel, a little bit. I haven't met him before. And I thought I would just start by discussing just a little bit about my background because, as I stand up here, if you'll pardon the pun, you don't know me from Adam. And I'm in the south of the States, and you're looking at me and you're probably thinking, oh yeah, great. The evolutionist, long hair, yeah, figured, right? Some long-haired hippie going to talk about evolution, right? Probably votes Democrat. Probably eats tofu. All those kinds of things. Well, sometimes things are not as they seem. First thing I'll tell you is that I'm a family man. I'm supported by a wonderful wife named Val and my children. This picture's a little bit old, but it shows you who they are. I love my family. I love my children. I love the place that God has planted me. This is British Columbia, Canada. That's where I come from. I live near Vancouver, British Columbia, and I teach at Trinity Western University, which is near there. We have been blessed to get a small piece of property on one of the islands off the coast of British Columbia, and I am constantly amazed at the wonder of God's creation when I go out there. So don't worry, we couldn't afford waterfront property. That's not my water, not that, yeah. I'm also a bit of a redneck, and unapologetically so. I come from the northern part of my province, and I grew up hunting and fishing and those types of things, so no, I don't eat tofu. Here's me on our property, clearing some of the land, and we're in the process of building a small cabin there. So I love God's creation. And if you look closely, it'll have to be in the front for people to see it. There's actually a deer looking at me running the chainsaw there. And what's interesting is on this island, the deer will actually come to the sound of the chainsaw because they love to nibble on the tops of the treetops, the limbs, once they've been felled. So I also enjoy that island for its opportunities in the fall. Like I said, I don't eat tofu. I'm a hunter, and I like hunting those deer. I'm also, and this is where most people in this conversation know me from, I'm also an evolutionary creationist. I hold that evolution is the mechanism that God chose and used to bring about biodiversity on Earth. So here's a photo of me doing a lecture a number of years ago, and if you can't see it, but the title says on the slide behind me says, why I am an evolutionary creationist. So even though you've seen some things about my personal life and who I am, the most important thing about me is that I'm a follower and lover of Jesus Christ, that I name him as my Lord and Savior. And like Nathaniel, we both have a passion for exploring God's creation and using the tools of science to better understand that creation. So that's part of what I'm going to present to you today. So there you go. But yeah, I have long hair. I get it. I saw one other gentleman here with long hair, so I felt at least somewhat affirmed. Okay. Here's a quote from Abraham Kuyper that makes a point that I would like to make, and it makes it more strongly and better than I could. He says, "There's not 1 square inch in the whole domain of our human existence over which Christ, who is sovereign over all, does not cry, mine." So is science an acceptable activity for a Christian? I would say absolutely, because if every square inch of creation is Christ's domain, then it is an act of worship to explore that creation. Here's another very famous quote from Francis Bacon. He says, "Let no man think or maintain that a man can search too far or be too well studied in the book of God's word or in the book of God's works." So the idea that God has given us 2 books, the creation that we can see as a form of general revelation, but also God's scripture that he's given to us as special revelation. Now, if we think about these 2 forms of revelation, it should become pretty clear pretty quickly that there can be no inherent conflict between these 2 forms of revelation. If God is indeed the author of both, then there cannot be an inherent conflict between those 2 forms of revelation. What we read in Scripture and what we read through science should cohere. They should be connected to one another because they have the same author. Now, wouldn't it be lovely, I could say, and there's my last slide and we're done, right? No. The conversation that we're in, of course, deals with the issue that we appropriate science and we appropriate Scripture through human activities. So we have the process of exegesis and hermeneutics to better understand God's Word, and we can use the scientific method to better understand God's world. Now, it is probable, even likely, I would say, that in the process, because both exegesis and science are human activities, and because humans are involved, therefore they are both flawed activities, we can expect that there may be apparent conflicts between what we see in Scripture and what we see in science. But the point to make is this: the problem is not God's problem. The problem is a human problem, because both of our exegesis and our hermeneutics and our science are human activities. Okay. So, despite the possibility of apparent conflict between Scripture and science, both are sources of God's revelation. And Christians aren't well served if we denigrate either form of revelation. We should have the ability to do robust science and robust exegesis and hermeneutics with the expectation that these will cohere, even if we have to live in the tension, perhaps in a given time, where we don't necessarily see the full connection between these two sources of revelation. Here's just one little quote from my own institution, from Trinity Western, our statement on creation. We say, "A biblical view of creation does not constrain legitimate scientific inquiry and research because we accept two sources of information, biblical revelation and natural revelation." Another way to put this, to say then, is that well-supported science is a witness. It's an imperfect witness, but it is a witness to God's revelation in the form of natural revelation. And actually, I don't have time to get into it here, but there's a very good argument to be made, a very good case to be made, that the reason why science has its flourishing in Christian Europe in the time and history that it does is because those early practitioners of science were convinced that they could figure out how God had ordered the cosmos, because they were convinced that there was a mind behind the cosmos and that there would be order and regularity to it because of that. So science in many ways has its motivations from a Christian theistic understanding of the world. Okay, let's talk a little bit about theory. Nathaniel introduced this well with some quotes from my book, so we don't have to spend a huge amount of time on this. A theory in science is something that's quite different from the colloquial usage of theory. If somebody says on Facebook, well, that's your theory, well, what are they saying? Well, that's your guess, that's your conjecture, that kind of thing. In science, a theory is an explanatory framework that has withstood repeated experimentation. So it makes hypotheses that then you can test, and then you can either accept, you can fail to reject the hypothesis, or you can reject the hypothesis. But in colloquial usage, as we mentioned, theory means something closer to guess or conjecture. To put it another way, only a theory is actually pretty high praise from a scientific viewpoint. So if somebody says, well, that's only a theory, the intent is to try to denigrate it using sort of that colloquial usage. But in fact, to a scientist, we perk right up and you say, oh, theory. That means that it's a well-tested explanatory framework that makes accurate predictions. Okay, so I put this comic here just to sort of ease the shock of what I'm going to say to you now. It's a fantastic comic. Maybe fishy crackers actually argue over their origins as well. Hopefully they can come to some understanding that they're still, that they're brother and brothers in Christ and that they don't have to argue about it. But they say, despite what you may have been told as a Christian, evolution is actually a theory in that scientific sense. It's a well-tested explanatory framework. It's supported by a very large body of experimental evidence. I could put each and every one of you to sleep if we extended this lecture as I went through the reams of evidence that we have in support of evolution, because it's been a productive scientific theory for 150 years, which is not to say that I might not put some of you to sleep. I hope not to, but hopefully not. It makes accurate predictions, and we have not yet falsified it through experimentation. But the idea is that all scientists hold theories somewhat provisionally. You can have a very well-supported theory. It can make boringly accurate predictions. But we always hold it at least somewhat tentatively, because we know that future experimentation may cause us to revisit our ideas. Although in that case, any addition or modification to a theory still has to explain the data that has been previously accumulated. Okay, so evolution is also the most foundational theory in biology. Here's a very famous quote from a population geneticist who happened to be an Orthodox Christian as well. He says, "Nothing in biology makes sense except in the light of evolution." Okay, so Darwin had a hypothesis at the beginning. Evolution there was a time when evolution was a hypothesis. His idea was that species share common ancestors. So here he mused that perhaps populations might become separated, and as we've heard, Darwin didn't have any idea about genetics. He had some idea about heritable variation. But we would now say populations become genetically separated in some way. This means that genetic changes through mutation, recombination, and so on would not be averaged across those two populations because they can no longer exchange genetic information with one another. This can allow for differences between these two populations to accumulate and their average characteristics to change. Now, these differences may lead to new species over time. And this process is actually much like the formation of languages. So in the book, Adam and the Genome, I spend a good deal of time drawing an analogy between how languages change over time and how species change over time. And actually, I should make a quick aside just to say that nothing I've said so far is especially controversial even from a young earth creationist perspective, because young earth creationism has a large amount of speciation within it, as Dr. Jeanson has been discussing. So how do new languages form over time? Well, again, populations may become separated, a common ancestral population of speakers. We know through experience that languages have this incremental process of change within them as well. Changes in syntax, changing in spellings, perhaps neologisms that are coined in a new population. And if you have two populations that are now separate, they're not averaging these differences across them. So again, we can have differences accumulate, average characteristics can change, and these differences may lead to new languages over time. Now, it doesn't necessitate that you will have new languages form, but it at least leads to that possibility. So you can think of species and languages as information systems, transmitting their information from one generation to the next. But both of these systems, species or languages, do not transmit that information perfectly from one generation to another. So there's this possibility of change. Okay, here's just one example that I'll show as a way to sort of get our mind around how this works. If I put this up and allow you to read it, most of us, the vast majority of us in this room, would not be able to read this. This is actually Anglo-Saxon that you're looking at from about the year 990. And this is John 1:29 from the West Saxon Gospels. Now, I love to use this as an example because it's got Middle Earth in it, right? Here is God's Lamb. Here is something along the lines of the doing away of Middle Earth's sin, which if your memory verses and you know what John 1:29 is. Okay, here's the Wycliffe Bible from the 1300s. Now, this is a lot more comprehensible to us. It's closer to us, but it still shows some affinity with Anglo-Saxon. Here's the Tyndale New Testament in the 1500s. Here's the King James Version from the 1611. One of the reasons why this analogy works so well for Christians is because we're all familiar with language change over time because we read different editions or different translations of the scriptures. So if you've read a King James Bible and you've read a modern translation, you know that languages can shift over time. And here's there's the present-day King James. One of the ironies of the King James only movement, of course, is that they don't use the 1611 King James. They use the Cambridge editions of the King James. But that's what now I've gone from preaching to meddling, right? So we don't want to do that. Okay, and here we have a modern translation with the NIV. Now, the point is that it's not like everybody on one day in 1394 sat down and said, we've been speaking Anglo-Saxon for a few hundred years, it's time to change. What we're looking at is actually snapshots that are taken from a continuous process over time. It's also decidedly difficult to draw a line of demarcation on this process to say, okay, who was the first speaker of modern English? Well, it's an incremental process over time. It's a gradient, and it's decidedly difficult to draw a line on that gradient. Okay. You can think of this as sort of a fossil record, for example. If we had this, the West Saxon Gospels, and then we had the modern translations, we might look at that in the present day and say, wow, there's a huge difference between those two, which suggests to us that these are separate entities, even though there's similarities that we see between them. But Because we have these other snapshots, we can see that there is a plausible transition from one to the other. And actually, historically, we know this. Every generation spoke the same language as their parents and as their children, all the way along through this process. Yet what we see over a long period of time is an incremental process of change where a large amount of change accumulates in a particular lineage. Okay, so too with species. The shift in average characteristics over time, either of a language or of a species, means that we have separate populations that either potentially could become new species or they could become new languages, but the process is a gradient over time. Okay, so that was Darwin's original hypothesis, and that's a bit of an analogy to help us understand how that might work, although, as I'll come back to in the second lecture, that population level understanding is quite important as well. Species form as populations shift their average characteristics over time. New languages aren't formed by two people that go off and start their own language. We're talking about population groups that shift their average characteristics if they diverge from one another. We also, Nathaniel spoke a little bit about whale, some of the evidence for whale evolution. And this is one example that I like to use as well. And the idea is that evolution can sometimes force you into some rather counterintuitive but also intriguing predictions. So if we look at present-day whales, we wouldn't have, and just looked at present-day whales, we wouldn't have this idea that instantly came to mind that they're the descendants of four-limbed terrestrial tetrapods, four-limbed organisms. They're so exquisitely adapted to the aquatic environment. Darwin actually got completely lambasted for this as a prediction in the original edition of The Origin of Species, and a Christian apologist at the time actually takes him to task on that, and I discuss it a little bit in the book. Darwin was so embarrassed by that pushback, that how he could, propose such an outlandish idea that whales are the descendants of four-limbed terrestrial ancestors, that he actually greatly reduced that section of The Origin of Species in the second edition because it was so embarrassing to him. Now, Darwin was a man ahead of his time on this point, but the lines of evidence that I'll show you now were not available at that time. Okay, so evolution can produce a hypothesis that modern whales, or cetaceans as they're called, are descended from ancestral terrestrial organisms through a series of transitional forms. This makes certain predictions about what one might find if one looked in the fossil record, and Dr. Jeanson showed some of these species. But the point to make is that we fail to reject the hypothesis of common ancestry by what we observe in the fossil record. If we look in the fossil record, what we see are a number of species that are not in the present day and are unlike anything that we see in the present day, and blur the distinction between a terrestrial four-limbed tetrapod and a fully aquatic modern whale. Some of these species were actually known at the time of Darwin. The basilosaurids is one, and these are ones that have very, very small hind limbs that are still outside the body wall. So they actually do have hind limbs that are outside of their bodies. That's not present in present-day whales. Present-day whales have a little bit of bone back there, but it's not outside. It's encased within the body wall. The fossils that were known back at Darwin's time of the basilosaurids they weren't instantly recognized as mammals, hence the name basilosaurid, which sounds a lot like a dinosaur. They were miscategorized at first. And also, the samples that they had were not well preserved enough to preserve those small hind limbs that are present. Now, those hind limbs are not capable of bearing the body weight of those large animals. These are aquatic animals, but they have these tiny hind limbs and well-established front flippers. So we fail to reject this hypothesis when we look at the fossil record. Now, another prediction that might arise is that it's possible, if you look at the embryology, the early embryological development of these species, that we might see some hints of a prior past. Now, evolution doesn't guarantee that that's the case, but it's a possibility because embryological development is often difficult to change even over long spans of time. So embryologically, we often see hints of other histories when we look at present-day organisms. So it's a chance or a possibility that whales might display some characteristics in their embryological development that are reminiscent of a four-limbed terrestrial past. So the first thing to say one way to test that hypothesis is just to look at present-day cetaceans and their embryological development. So what we have here are early dolphin embryos, and they have several features of their development that are challenging to explain from a non-evolutionary perspective. The first is that modern-day whales are actually four-limbed organisms, but they are four-limbed organisms only for a very brief period of their embryological development. So you've seen that in present-day whales, in the adult form, they have a little bit of bone back there, but it doesn't extend beyond the body wall. Early on in embryological development, whales actually do have four limbs. So the developmental program that says make a four-limb bud runs, as it does normally in all four-limbed animals. The developmental program that says make a hind limb bud also runs in whales, just as it does in other mammals. But later on in development, a second developmental program actually comes along and overrides the developmental program in the hind limb bud only. So basically, biologically, what happens is start a four-limb bud, start a hind limb bud, and then some hours later, another program comes along and says, no, we don't want to do that. Come on back in. We don't want to do that. And it's actually at the molecular level, we've investigated this, and it's a second developmental program which overrides the first. But the first one runs normally at the beginning of embryological development. Another interesting feature about whales is that, like any good mammal, they start off with two nostrils on the front of their face. So one of the defining characteristics of mammals is that they have two nostrils, and we have them on the front of our face. The difference, of course, in whales is that they don't have two nostrils on the front of their face in their adult form. They have a blowhole on the top of their head. Developmentally, whales start off just like any other mammal by having two nostrils on the front of their face. And you can see the image here. And only later in development do we see movement of that nostril, those nostrils, to the top of the head. So developmentally, again, similar pattern. You start off in the way you've always done things, but then possibility for modification later. Okay. Now, I'm a geneticist, and of course, one of the most compelling lines of evidence that we'll talk about in some detail in this conference is the lines of genetic evidence that support the hypothesis that different species share common ancestors. So one thing you can do is you can look in present-day whales, as an example, and to look to see if they have any remnants of genes that show a dedication or an adaptation to a terrestrial manner of life. And the answer, or the result here, is that they do. So one category of genes that all mammals have are these things called olfactory receptors. So these are genes that are turned into proteins. So it's a DNA sequence that encodes for a protein, a large number of these different kinds of proteins. These proteins are put on the surface of our nose, so on the nasal epithelium. We breathe in air, and different chemicals in the air that we breathe bind onto these proteins, change their shape, and that change in shape is eventually read out as changes in nervous system activity. So this is how mammals have a sense of smell. And mammals, as a rule, have about 1,000 genes in their genomes that are dedicated to this sense. And when we only have about 22 to 25,000 genes in total, it shows you that we have a very large percentage of our genes that are devoted to this particular sense. The sense of smell is incredibly important to most mammals. Now, the sense of smell is not important to whales in the same way, especially the toothed whales. There's some evidence that baleen whales may still have a little bit of a sense of smell, but toothed whales, or odontocete whales, so think of like killer whales, sperm whales, that sort of thing, they don't actually even have an olfactory organ. They just have a blowhole that goes straight from their body wall to their lungs without an olfactory organ associated with that. Now, the reason for this is pretty straightforward. Odontocete whales, toothed whales, don't use a sense of smell for hunting. If you've ever gone swimming and attempted to smell underwater, you know why that's not a viable strategy, right? But what's interesting is we can look in the genomes of present-day toothed whales, and they have the same olfactory receptors for air-based olfaction that any good mammal should have, except they have mutations that have accumulated in those genes that prevent those genes from being used. So whales have the genes for air-based olfaction. The other example here is that they also have the genes for air-based vision, for certain wavelength pigments that allow them to see effectively through air. Now, How do toothed whales do their hunting? They don't use the vision. They don't use vision primarily, and they don't use smell because they're holding their breath. They use echolocation. So even though they have shifted their manner of life to a decidedly different way of doing things, their genomes still retain the features of that earlier past that geneticists can see and observe. Okay, so what we see here then is converging lines of evidence, and scientists are always happy when they get converging lines of evidence from different disciplines. So we can look at DNA sequencing, we can look at embryology, we can look at the fossil record, and we can look at comparative anatomy and physiology, which was what sort of set us on this path in the first instance, because whales are mammals and share that common physiology with other mammals. So as a result of looking at all these different areas, we can see that the lines of evidence converge with one another. They support the same hypothesis, that whales are the descendants of terrestrial ancestors through a series of transitional forms. Okay, so as far as we know, whales don't lose much sleep over the idea that perhaps they descend from those grubby little land-dwelling creatures that we would never want to be. So, we don't know if they lose sleep or not. As Christians, we do sometimes lose sleep over this question. We want to know, are we also a part of this pattern of speciation? Do humans also share ancestry with other forms of life? And the short answer is yes. The lines of evidence that I've just shown you, embryology, paleontology, the fossil record, and DNA sequencing, all similarly have a coherent pattern of evidence that support the idea that humans do participate in this process, that we are also the result of evolution. So one way to do this, and I won't look at the paleontological evidence and the embryological evidence, but it's there. Let's focus a little bit on genome sequencing, because that is something that has become recently a very powerful line of evidence that more Christians are becoming aware of. A friend of mine, Randy Isaac, who was mentioned by Nathaniel, once said in an article, he said, prior to DNA sequencing, Christians had some wiggle room on this topic, but it looks like DNA sequencing has really taken away that wiggle room because of the compelling nature of what we're seeing in the genomes. So modern genome sequencing allows us to compare the human genome with other organisms. And morphologically, we are most similar to other great apes. So even Linnaeus put us in primates as a category, based on shared morphology, shared anatomy, and physiology. And primate, is, we're at the top, as it were, according to Linnaeus' strategy. So evolution would naturally predict that we would share the closest DNA similarity with other great apes because we share the closest morphological similarity with them. So it's a natural prediction. The human and chimpanzee genomes, when you compare them at a DNA level, base pair by base pair, are over 95% identical one to another. One of the interesting things as a biologist that we found out is that it takes, that languages actually change much more rapidly than species do. So you can actually have quite a bit of morphological and physiological change between species based on quite a small amount of genetic difference, which is really quite interesting. I know that Tompkins has, just as an aside to Nathaniel, I know that Tompkins has published some papers that dispute that value, and perhaps we'll have a conversation about that later, but yeah. Not only do we have the same genes, we also have them in the same basic order. So this is the idea of synteny, the idea that not only do we have the same genes, but we have them in the same spatial pattern. And there's no a priori reason why separately created species would necessarily have to have their genes in separate places, in different orders. To put it another way, our two genomes are pretty much exactly what you would predict as slightly modified versions of an ancestral genome. There's a huge amount of correspondence between our two genomes. Here's a diagram that Nathaniel also showed. This is just showing you an overall pattern of DNA similarity, now with human chromosomes on the left, and chimpanzee, the equivalent chimpanzee chromosomes on the right. This is very early work done in the 1980s when we were just doing DNA staining of chromosomes to look at them sort of as overall morphology of chromosomes. We, of course, have now sequenced over all of this, and the impression that you get of the same structures in the same order is confirmed at the individual DNA level. Nathaniel also mentioned pseudogenes, and I've just been talking about pseudogenes in whales, olfactory receptor pseudogenes. One of the interesting things about primates is that we too have lost quite a large number of these olfactory receptor pseudogenes. And that's actually not such a bad thing because we live in pretty crowded conditions. And if we had the, olfactory acuity of, a wolf or something like that, we'd be able to, it's like, somebody had eggs for breakfast, or somebody needs to take a shower, or something along those lines. So humans and primates in general have lost a large number of these olfactory receptor genes. Now, sometimes when I'm speaking to non-specialist audiences, one of the questions that comes up is to say, well, how do you know what you're looking at is actually a defective gene? Could it be something else that has another function that you don't know about yet? We'll get into some of the details, but one way to start thinking about this is to look at this image that I have here. So here we have a structure that if you were walking across a field and you came to this, you would know a few things about it. The first thing you would know is that, a building was here. There's a building here. But this building doesn't have all of the features associated with it that make it a fully functional building. It's lacking its roof. There's grass growing up through the floor. And at the genetic level, it's similar, it's analogous to what a geneticist can see. You can see that perhaps most or almost all of a gene is there in the genome, and you can see that certain mutations have occurred that render its function, render it not able to function in the same way. This is also further bolstered by the idea that we can actually do DNA comparisons between a large number of related organisms. So in many cases, we'll see that same gene, and it'll be functional in a number of species, but then it'll be non-functional in another species that's part of that overall species group. And as an aside, many times those different groups correspond to what a young Earth creationist would agree descend from one another, that they would actually be part of a Barraman. So in some cases, although this is interesting to me in following the young Earth creationist literature, we don't often see an example of using pseudogenes or nested hierarchies of genetic mutations as a way to define Barramans, to define different created kinds. I suspect that part of the reason for that is because these same lines of evidence also include humans in the primate Barraman, but that's something that we can talk about later. It's actually something I don't exactly know, and maybe I can learn more from Dr. Jeanson on that front. Okay, you're not going to be able to read this. But perhaps if you look at it later online, you can have a look at it, or it's in the book as well. So what you're looking at here are a large number of different olfactory receptor genes in primates. And if you could see the text, what you would notice is that not only do the same mutations show up in different species, but those mutations, or those different species, not only have mutations in the different genes, but many of them have exactly the same DNA letter change in each of those different species. So the question that you then begin to wonder as a biologist is to say, are we looking at independent mutation events that independently produce these different identical mutations in multiple species? Or is it a case where we've had a mutation take place which then has been inherited by a number of species as they've gone their separate ways? And we end up seeing these different mutations with identical characteristics that form these nested hierarchies. Just as one example here, so this is the same data set but now put onto a phylogeny. We see some cases where we have mutations that are specific to an individual species. So in this example, there are 15 that are human-specific, 4 that are chimpanzee-specific, and so on. We also see 3 in this little subset that are identical between humans and chimps. We also see a further 3 that are identical between humans, chimps, and gorillas, and a further 6 that are shared by humans, chimps, gorillas, and orangutans. But notice what we also don't see. We don't see this example in this data set, a case where we see the same mutation in humans and we see it in, say, orangutans, where we don't also see it in chimpanzees and gorillas. And the reason for that is if there's a mutation that's shared between humans and orangutans, under the evolutionary model, that mutation would have occurred in a population prior to humans and orangutans going their separate ways. That population is also the founding population of what will later go on to diversify to become gorillas and chimpanzees as well. So if we see it in humans and orangutans, we can predict with great confidence that it will also be present in gorillas and chimpanzees. And we do. We see this nested pattern. Okay, couldn't pseudogenes be in fact functional genes? Let's just dig into this a little bit more deeply. And again, it's going to be challenging to see this on the small screens, but we'll do the best that we can. So here's an example from the human genome of a number of genes on a chromosome. And there's one gene in the middle that is a pseudogene. So it's picked up a number of mutations that render it non-functional. Now, interestingly, this same arrangement of these same genes is also present in the mouse genome. So again, you won't be able to see it unless you're looking at it fairly close up. But we have the same order of genes on the mouse chromosome as we do on the human chromosome. Now, these genes do not have the same function. They're not in a sort of coordinated set of genes that have to be in this particular pattern in order to accomplish their functions. They could be in different places in the genome if they wanted to. The idea here is that we're looking at a small segment of chromosome where things have not been broken up and shifted since humans and mice shared a common ancestor at about 125 million years ago in the past or so. So we see in the mouse genome, we see that this gene is a functional gene. Its actual function is a nuclear hormone receptor. And we know from its structure, by looking at its structure, and also by experimental analysis in the mouse, that it actually has this function, because we can test it directly in the mouse. Then we see that same sequence, or something that is almost identical to that exact same sequence, in the human genome, and we see it in the same context among the same genes that we see in the mouse genome, but that gene in the human genome has some mutations in it which render it non-functional. Now, let's just explore a special independent creation model here for a moment, just to give you an idea of how biologists look at this data and maybe wrestle with these different ideas. And I can wrestle with this as a Christian biologist because, to me, whether God used evolution or did special independent creation, I'm willing to explore the evidence and see where the evidence goes. I'm good either way, because whether God uses what we would call a natural mechanism to create or uses divine special creation, God uses natural mechanisms all the time. God uses special stuff all the time. I'm good either way. Let's just go with where the data goes. What I would end up having to postulate as a Christian biologist is that, okay, there's a function here in the human genome. Say, okay, maybe this isn't a pseudogene. Maybe this is actually something that's functional. I don't understand what its function is, but I'll go on faith that there's function there. Perhaps I can test that later. And I'd have to say, okay, well, God intended that function to be there. And for some reason, it has to be that way such that it looks very, very much like a different function in mouse, because those two things cannot have the same function. I know that by looking at their comparative differences. So could God have given humans that function in another way? Yes, God is perfectly capable of designing that function into our genome in any location he chooses and in any way he chooses. So you end up with this interesting situation where you would have to say, okay, is somehow God constrained to put that particular thing right there to provide that function? Or am I looking at the results of common ancestry? And biologists, Christian biologists included, overwhelmingly go with, it looks like common ancestry. Okay, and this is just because geneticists get bored and they geek out and go digging around in databases. And this is just to show you, again, you won't be able to see it, but this exact same mutation that we see in humans that render this particular gene non-functional, that particular mutation, many of those mutations are also shared by chimpanzees as well. So even if we sort of solve the problem for the human genome, we would also have to discuss about why that function is there in that particular form in the chimpanzee genome. Okay, here's one, example that I also like to talk about. And it's in the book, this idea of Vitellogenin pseudogenes in marsupials and placentals. So Vitellogenins are this interesting category of genes that egg-laying organisms use for their biology. The way egg-laying organisms supply their embryos with a store of nutrition is with using these genes. They're very large genes. They're made in the liver of the mother and then transported through the blood to the developing egg. And they're laid up in the egg as a store of nutrition. So most of what you see in an egg yolk, that yellow stuff, a large percentage of that are these things called Vitellogenins. And the reason for that is the mother will eventually deposit an eggshell around that embryo, and then the connection between the mother and the offspring will be broken at that point. So the mother can't provide that material, nutrition to the embryo anymore. Now, placental mammals and marsupial mammals do things differently. We maintain a connection with our embryos through the placenta, if we're placental mammals, or we have marsupial mammals have a slightly different placenta that they use. So what this research group was interested in was to simply say, I wonder if we could look in the genomes of placental mammals and marsupial mammals for remnants of genes that are devoted to egg yolk production. So they wanted to know, are there still remnants of Vitellogenin gene sequences in marsupials and placentals? And the short answer is that yes. So other lines of evidence that I don't have time to talk to you about right now suggest that placental mammals, such as humans, share a common ancestor with egg-laying organisms at about 310 million years ago. If you look in the human genome, the way this research group answered this question was they said, okay, let's look in the chicken genome for where we find functional Vitellogenin genes. And then they noted what other genes are next to those genes in the human genome, or in the chicken genome. And then they went to the human genome and said, are these other neighboring genes still around in the human genome? Are they present? And they are in the same spatial pattern. And when you look then at the relevant spots in the human genome, wherever you see these little black bars, and this is not to scale because if I drew it to scale it would be very difficult to see, but the general impression is the right one. Wherever you see a black bar, you've got sequence matches between the human genome and the chicken genome. So there are small fragmentary remains of Vitellogenin-like sequences in the human genome at precisely the location that you would predict them to be based on the flanking gene sequences that we see in chickens. This same pattern is also present in opossums as well. So opossums, just as one example of a marsupial, and they actually have even better retention of these sequences. So again, one could try to make the argument that there needed to be a function that's different from Vitellogenin functions that has to be in the genome of the opossum and the human at this specific location, or you could say, this is evidence that supports common ancestry. Okay, and this is just because biologists get bored, and yes, those same mutations are shared by a large number of different species, placental, marsupial, and otherwise. Okay, I think based on time I'm going to skip over this, but we can come back to it if need be. Sometimes folks are curious about, evolution is good at removing function, but where does new information come from in evolution? Is evolution capable of producing new information? And the short answer is yes. I've got a big section in the book that goes along these lines. Here's just one example where we can look at closely related organisms. And what we see, so here's humans, chimps, and gorillas again. Sometimes when we look through the human genome, we'll find genes in the human genome that appear to be functional. And we will notice that those genes aren't present as genes in our closest relatives, in gorillas and chimpanzees. But when we carefully examine the DNA sequences of those other species, we find out that there are sequences there that are almost identical to the functional gene in humans, but are just perhaps 1 or 2 or 3 mutations away from becoming a gene, as it were. And the type of research, we have to be very, very careful that we're not looking at loss here. So you have to go through all sorts of controls to make sure that we're not looking at something that was functional in all 3 organisms and then was lost in gorillas and chimps. But we've done this, and there are a number of genes now that fit this category where it seems like a gene has come into being in the genome through the result of mutation. Okay, let's see. Let's just briefly talk about design arguments before we land the plane here. Design arguments have a long history within Christianity. And in fact, one of the things I like to do as a hobby, scientists, we have weird hobbies, what can I say? One of the things I really like doing as a scientist is reading the apologetics literature from the 1600s. Well, why is that? Well, in the 1600s, the church went through a very similar discussion to the one that we're going through right now. Except the science of the day wasn't genetics or genomics. The science of the day was heliocentrism, whether or not the sun was the center of our solar system or whether or not the earth was the center and the sun went around the earth. And you can find fantastic apologetic resources from the 1600s that address this question. I tell you, I could not get away with a title like that. You gotta love the length of titles that people had back then, right? A demonstration of the existence and providence of God from the contemplation of the visible structure of the greater and lesser world in 2 parts, and then it goes on, and that's still all the title. My editor would not let me get away with that now. Okay. Here's an interesting case where this author, and I actually discuss his views in Adam and the Genome a bit on some other related issues. But here's a simple test case where he's actually talking about solar fusion. So he says, if it be asked, where is the fuel for those vast fires? He's talking about the sun, which continually burn. Whence is it that they are not spent and they are exhausted? How are those flames fed? So he has no idea how you can have a sun which is, on fire and on fire for a really long period of time without it being consumed. So he says, none can resolve these questions but the Almighty Creator, who bestowed on them their being, who made them great and wonderful, that in them we might read his existence, his power, and his providence. Okay, so in modern language, Edwards is making a design inference. He's saying there's no known causal explanation for the longevity of solar combustion. God can maintain solar combustion using miracles, or by adding fuel perhaps. So since there is no natural explanation, we can infer from solar combustion, to follow this individual, that God exists, and it's an inference to the best explanation. Now, Edwards is very clear that on what his sort of motivation for writing is. And just as an aside, this is not the American Puritan theologian Jonathan Edwards. Sometimes this gets confusing to people. This is John Edwards, who's an English apologist writing in the late 1600s. He says, if I cannot by this attempt convince atheists, yet I hope to do something toward preventing the spreading of that pernicious infection which they are the authors of. Or if my hopes fail me to this, I will not despair of confirming and strengthening such who are really persuaded of the doctrine here presented, or here treated of. So he's basically saying, if I can't at least take atheists down a peg or two, at least I can shore up the faith of Christians, right? Okay. So, now, what does he not know, right? What does Edwards not know? He doesn't know about solar fusion, and it's going to be quite a while before solar fusion is understood scientifically. Now, is the understanding that solar fusion is the mechanism by which the sun is kept burning for long periods of time, does that negate an understanding that God is the author of that process, that God is somehow using that process as a way? Like if we could go back in time and explain it to Edwards, would he have a crisis of faith? I hope not, right? Okay. So, are design arguments that rely on a lack of evidence a good idea for Christians to employ? I would say no. This is a quote that I like very much from Dietrich Bonhoeffer, and I'll just read it in part. He says, Weizsäcker's book, The Worldview of Physics, is still keeping me very busy. He's actually writing this from prison. How is it that Bonhoeffer, who is in prison, has the ability to interact with a book on the science-faith dialogue at his time and place? He is an example to us all. He says, the worldview of physics is still keeping me very busy. It has again brought home to me quite clearly how wrong it is to use God as a stopgap for the incompleteness of our knowledge. If in fact the frontiers of knowledge are being pushed back, and this is bound to be the case, then God is being pushed back with them, and therefore is in continual retreat. We are to find God in what we know, not in what we don't know. God wants us to realize his presence not in unsolved problems, but in those that are solved. Okay. One way of thinking about this is it's very common for Christians to view evolution as somehow against design or contrary to design. That God is either a designer and a creator. We could substitute creation for design if we wanted to there. We'd say evolution versus creation, evolution versus design. I take a different view. I actually take a view that evolution is the design, that evolution is a process that God has ordained and sustained and uses providentially to bring about the biodiversity on Earth that he desires to be there. So it's a different way of thinking about things. Okay, Nathaniel sort of stole my thunder here, but that's okay. This is from Todd Wood. This is a quote, and I'll finish with this quote here. This is Todd, a few years ago, talking about the truth about evolution. He says, I hope this doesn't turn into a rant, but it might. You've been warned. He says, evolution is not a theory in crisis. It's not teetering on the verge of collapse. It has not failed as a scientific explanation. There is evidence for evolution, gobs and gobs of it. It is not just speculation or a faith choice or an assumption or a religion. It is a productive framework for lots of biological research, and it has amazing explanatory power. There is no conspiracy to hide the truth about the failure of evolution. There really has been no failure of evolution as a scientific theory. It works, and it works well. I say these things not because I'm crazy or because I've converted to evolution. So as Nathaniel mentioned, Todd Wood is a young Earth creationist. Nevertheless, he holds these views. As you might expect, he gets a bit of flack for it. He says, I say these things not because I'm crazy or because I've converted to evolution. I say these things because they are true. Then he goes on to say, I'm motivated because I've read some other clueless person saying that evolution is a failure. But you can read that on his blog if you're interested. All right, with that, I'll thank you for your attention. And I guess questions are after dinner, but thank you very much for your attention. Okay, good. It's a delight to be back, and hope you enjoyed your dinner, as did I. Is there something about Chick-fil-A that just is part of the Southern experience, or? We don't have Chick-fil-A where I come from, so hopefully the Vitellogenin discussion didn't create any ethical concerns for you as you enjoyed your chicken sandwich. Okay, in the last talk, I discussed the heliocentrism issue. So what I'd like to do is actually return to that a little bit as a way to think about how Christians in the past have negotiated some of these sorts of issues, similar to what we are facing now. I sometimes tell my students, lucky you get to live at a time when the church gets to wrestle with these sorts of serious questions. We haven't done this for a couple of 100 years. And they say, I don't want to live in a time when the church wrestles with these kinds of questions. I want to live in a time when the answers are there for me. But such as it is, God has appointed that we live in this time and in this place, and this is the time that we are given. And it is a time when the church is wrestling through these things. Just as an aside, though, I think that as long as the church can learn to dialogue graciously on these points to the best of our ability, and we're not perfect, we're humans, right? So we are going to make mistakes, absolutely. But to the best of our ability, if we can dialogue graciously on these issues, at the end of the day, whether one accepts one version or the other, if we recognize one another as brothers and sisters in Christ, and then if the world can see that we can dialogue charitably on these things, then it won't become a missional problem. It won't be something that people on the outside look in and say, oh, look at those Christians fighting over these things. We're meant to be known for our love, and this is an opportunity where we can show love. Now, I understand that, for the different denominations and whatnot, can be quite a struggle in certain areas. I don't diminish that. But if we can do it in a charitable way, I think if we can learn from history on that point, we'll be ahead of the curve on this one. Okay, let's move back to something that is not theologically challenging for most of us in the present day. So if we look back in history, we can see that the idea that our solar system, and even to use the word solar system is to imply sort of a Copernican way of looking at our universe. Nicholas Copernicus was the first individual to propose sort of a coherent mathematical model of a heliocentric solar system, and you can see when he lived. So up until that time, the view of how the universe was constructed was not viewed to be heliocentric. The notion was that the Earth was at the center of the universe. It was a geocentric or Earth-centered system, and everything went around the Earth. So Copernicus's idea that the sun might be the center was viewed as something that was a new sort of mathematical way of thinking about things. And it kind of came onto the radar of the church as well, that perhaps there was, something theological here that might be concerning. So this is a very famous quote from Luther. Now, I don't want to press Luther too far on this particular point, because Luther wasn't a scientist, and as far as we know, this was kind of just like an off-the-side, kind of off-the-cuff kind of comment. So this isn't perhaps as he's had opportunity to reflect deeply on this, but he says there's talk of a new astrologer. So astrology back then, that's actually not a bad way to... astrologer now doesn't mean what it meant back then. So there's a new astronomer who wants to prove that the Earth moves and goes around instead of the sky, the sun, and the moon, just as if somebody were moving on a carriage or ship might hold that he was sitting still and at rest while the Earth and the trees walked and moved. But that is how things are nowadays. When a man wishes to be clever, he must invent something special, and the way he does it needs to be the best. The fool wants to turn the whole art of astronomy upside down. However, as Holy Scripture tells us, so did Joshua bid the sun to stand still, and not the earth. Copernicus didn't really encounter too much resistance. And again, there's a lot of history here that makes for very fascinating reading, but one of the things that we can see in the response to Galileo, who was the next individual to come along and sort of further this line of investigation in a major way, we can see in the responses to Galileo that Copernicus's views weren't seen as particularly theologically threatening because it was thought that he was just sort of proposing a hypothetical model, kind of a mathematical model to describe how, a solar system might work mathematically. But it wasn't viewed as particularly threatening because it wasn't seen to have much traction in reality. So it's a nice model for the philosophers and the mathematicians, but it doesn't really work that way. Well, Galileo comes along in the late 1600s, and it's actually remarkable to think that we're only about 400 years removed from that time. It just shows you how quickly science advances since that time. And Galileo actually had some observational evidence that he could bring to the table. So he was one of the first individuals to construct and use a reasonably high-powered telescope. And with that, he observed the moons of Jupiter, and he also observed that Venus has phases. Actually, I was on a hunting trip many years ago in the north of British Columbia, and it was a nice winter's night, and I was sitting around the campfire with a bunch of guys after a day's hunting. And if you're a hunter, you might know that, sometimes people have spotting scopes, which are basically low-power telescopes. And I happened to notice that Jupiter was visible, and I said, we should set up Gary's spotting scope and have a look at Jupiter, because I bet you we can see the moons. So we did, and we had this beautiful experience of looking at the moons of Jupiter, and then we had this fantastic conversation about science and faith. And not everybody in the hunting party was, of the same mind as me, and I became painfully aware that there were many firearms around the location at the time. No, it all worked out fine. It was an example to have charitable dialogue, and also to appreciate God's creation. But what Galileo saw as he looked at Jupiter was that it has small moons that go around it. So here we have, and we still call them the Galilean moons to this day, the 4 that are visible in that manner. And what that did was it sort of provided in microcosm what a solar system might look like. Here we have a body, at a center, and we have things going around that. So could that potentially be a sun, like a model of what the sun and the earth look like? The other thing that Galileo observed, one other thing that I don't have on the slide here, is that he saw that the moon had mountains on it, that the moon was not a uniform sphere. And up until that point, everybody thought that everything celestial was perfect in the sense that it was a perfect sphere. So that was theologically troubling as well. I know it seems a little bit odd to think about these points of theology that don't have any traction with us, but they had quite a bit of traction back then. The other thing he observed was that Venus has phases, just like our moon does. So Venus waxes and wanes. Venus will appear at sometimes as a full Venus and as a crescent Venus. Now, that is something that cannot be squared with the previous model of geocentrism that was prevalent at that time in that place. So this observation pretty much ruled out Ptolemaic geocentrism, but an additional model was also developed at this time by Tycho Brahe, and he had a different model. So this is a model wherein the Earth is still the unmoving center of the system, but the sun and a few of the planets, the sun is the center of a few of the planets, but then the Earth is still the unmoving center of the entire system. Brahe's model is basically the idea that if you could reach in from the outside and grab the Earth and hold it still and let everything else go whirling around it, that's the mathematical model that Brahe was proposing. And there's actually no way to distinguish between those two models, or there wasn't at the time technologically. But it was theologically a more satisfying model because of the theological commitment to an immobile Earth. So what we needed was a different point of view, and we needed a different point of view for the Earth itself. And this has to do with something called stellar parallax, this idea that if the Earth actually was moving through space, we should be able to see this particular effect when we look at stars that are close to us relative to stars that are far away. So I sometimes get my classes to do this, so I'm going to get you guys to do this too. If you want to observe parallax, take your hands, put your fingers up with me. If everybody does this, then I won't feel as foolish. There you go, although I'm the only one on the live stream, right? Okay, so put one close to your face, and then put another one in line farther away. Now close one eye, and then wiggle your head back and forth. you can see that the relative position of your two fingers seem to switch places with one another. So what was expected was if the Earth is going around the sun, that our perspective would shift. So a nearby star and a faraway star, as the Earth is going around the sun, we should be able to see the relative position of stars shifting to one another. And that would be very good evidence that the Earth was in fact in motion and not the sun. So the idea was plain, and it was easy to see how it could be tested, but technologically there was no way to do this back in the 1600s, just because you need very high magnification, powerful telescopes in order to do this. There wasn't an appreciation for just how far away the stars are. Can you imagine trying to detect parallax if I held two fingers together like this and then I stood across the street and you wiggled your head back and forth a bit? You wouldn't be able to see it. Now, you might be able to see it if you had a very high power telescope and very precise ways of referencing, but it wasn't accessible to them at this time. Now, the inability so we were able to observe this in the 1800s, and there were actually other convincing lines of evidence that came in prior to that time as well. But the inability of scientists to observe this stellar parallax in the 1600s was taken as confirmation that the Copernican opinion, as it was called, was not only unscriptural but also poor science. So there was this expectation this observation should be made, and it was felt that that the tests had been done and the observations had failed. So let's go back to John Edwards from the 1600s. I quoted him on solar fusion. Well, he didn't know he was talking about solar fusion, but we quoted him previously. So here's John Edwards, 1696, discussing theologically what the issues might be with respect to heliocentrism versus geocentrism. So he says, the Copernican opinion seems to confront a higher principle than that of reason. So languages have shifted a bit, so it's going to take a little bit of translation as we go through, but of course languages shifting was something that we talked about previously as an example. But he's basically saying there's a higher principle to that of science or reason. And when the chips are down, you go with the Bible. You don't go with science. That's basically what he's saying. He says, if we will speak like men of religions and as such own the Bible, so own, like do you own it, as in do you stand on its authority? We must acknowledge that their assertion is against the plain history of the Holy Book. For there we read that the sun stood still in Joshua's time and went back in King Hezekiah's. Now, and this is the same passage that Luther was referencing as well. Now, this relation is either true or false. If it be the latter, then inspired scripture is false. So what Edwards is willing to say is, if you can prove to me that heliocentrism is true, then that indicates to me that scripture is false. It says, if it be the former, i.e., if the relation really is true, then the sun hath a diurnal motion about the earth. For the sun standing still could not be strange and wonderful unless its general course was to move. So for Edwards, if you have to make a choice between science and scripture, between reason and the Bible, then you go with scripture, not with reason. Okay, now, that's obviously not the situation in the present day. Most of you are not losing sleep over the heliocentric nature of our solar system. But have you ever paused to wonder why you are a Copernican Christian? Or why, for me growing up, Darwin was a bad word at church. If you heard the word Darwin, that was bad. Evolution was a bad word. You don't talk about that. But Copernicus? No big deal. Copernican never read, if somebody called somebody a Copernican, not that anybody ever did, it wouldn't have registered as a bad thing. But have you ever wondered why that's the case? Interestingly, there is no scriptural support for heliocentrism. And if there was, we would have seen those texts brought to bear in the conversation that Galileo and others engaged. There simply is no scriptural support for heliocentrism at all. It's just not there. At best, you can argue that Scripture is silent on the issue. Now, I'm not 100%, I looked at some of Answers in Genesis' materials on this case, and I believe, I may be wrong, maybe Nathaniel can correct me, but I believe that this is the position that Answers in Genesis takes, that Scripture is simply silent on this issue. Although that is very much not what was thought to be the case back in the 1600s. They very much thought that the verses, one example there on the bottom from Ecclesiastes 1:5, they very much thought that these verses had a bearing on the discussion. So, How is it that we've made our peace with Copernicus? Perhaps you don't even know. Perhaps you didn't even realize that there might have been this issue to make peace with. Although, interestingly, there are still a very few Christians who do hold to a geocentrist view, and they can be found. So, for that matter, we've got NBA players that hold to a flat Earth, right? So, okay. So why is it that we're not troubled by Copernicus? In the present day, most Christians are perfectly comfortable with the idea that Scripture in some ways speaks to the original audience in a way that the original audience would have understood it, such that it would not have been a barrier to their understanding. So one way to think about this is that Scripture is speaking in a phenomenological manner. So we even say this now, the sun rises, the sun sets, as it were. Even though those things are scientifically false, as it were, the sun does not rise, the earth turns, the sun does not set, the earth continues to turn, and so on. But that's phenomenological language. It's from the perspective of what we observe, and it's not considered untruthful to speak about things in that way, right? Nobody says, Dennis, you're a liar, when you say that it's a beautiful sunset. Okay, so, and of course the analogy there to evolution is fairly straightforward. We should at least as Christians be open to the idea that perhaps Scripture is speaking in a phenomenological manner and speaking in a way that the original audience would understand. So when we hear in Genesis that God is stating that different species are being created or different organisms are being created according to their kind, and kind being defined as reproduction, it's not surprising that that would be a phenomenological way of describing things. The ancients would not have had an access or an understanding to larger spans of time to understand that perhaps species might shift over large periods of time. Okay, so the 1800s saw significant developments in biology, namely Darwin's theory of evolution by natural selection. Darwin was the first to provide good evidence that modern species are derived from ancestral species through a series of transitional forms, and we've seen in the previous lecture how that particular hypothesis continues to garner very strong support from modern scientific enterprise. Like heliocentrism, evolution has encountered resistance from within the Christian community. Evolution is fairly widely doubted among Christians. It doesn't take much effort to look at surveys of North Americans, find out what their opinions are on evolution, and this is a theory that is widely doubted by Christians. And most Christians feel that there's insufficient evidence to support it as a viable scientific theory, especially for human evolution, right? If you look at the different sort of Pew Forum survey results, there's usually more resistance or more reluctance to say that humans might be a part of this particular process. As we saw in the last lecture, though, modern-day genomics nicely fits with other lines of evidence that we've seen, and that evidence is quite substantial that humans do indeed share in this process of evolution, that we are the products of an evolutionary process. Now, what I'm going to speak to now is that not only did we evolve, not only did humans come about through, not only did God bring about humans through an evolutionary process, but that also took place as a population rather than starting from a single ancestral couple. Okay, so let's just briefly review. We had Darwin's ideas about him proposing that species share common ancestors, the idea that populations become genetically separated. We've got changes, so this is just review from the previous talk. We've got these different changes that perhaps can accumulate, giving a shift in average characteristics over time. The differences may lead to new species over time. And then we drew that analogy to languages, how languages shift over time, populations becoming separated, differences showing up, average characteristics changing. And we made the point that both languages and species are information systems that are copied imperfectly, so we have this opportunity for change. And then I showed you these texts to sort of illustrate this process using language as an example. Now, here's the point that I want to make here, is that languages don't start with two individuals heading off and starting their own language. Likewise, species don't start, typically, with two individuals going off and starting a new species. Or languages don't start when two individuals start suddenly speaking a different or a highly different version of the language that they spoke before. Species don't start when there's some sort of dramatic mutation that makes them different from the previous organisms that they were a part of. So evolution, like with languages, is all about average characteristics of populations shifting over time. Now, so the idea that any large mammal, us included, would have started as a population of just 2 solitary individuals would be extremely unlikely, even in the absence of any sort of genetic evidence. And that's because population shifting over time is how evolution works. It's a gradient. Okay. Now, I made this, I'm not sure if I showed you this slide or not, but one way that you can tell if species are connected with one another is we've sort of seen some of these lines of evidence in the previous talk. We can go around the world and easily find related languages. I use this example in the book. I grew up with one set of grandparents as Frisian speakers with a last name like Venema, or if you're Frisian, Venema. That it's not surprising that some of my ancestry comes from the Netherlands. And what was interesting to me as a child, I relate this somewhat in the book as well, is that I would notice my grandparents speaking West Frisian, and I would notice how uncannily similar some of those words were relative to English. And what I didn't know then, but I do know now, is that modern-day English speakers and modern-day West Frisian speakers, Friesland is a province of the Netherlands with its own distinct language, those 2 language groups share a common ancestral population of speakers that lived at about the year 400 on the continent, and then some of them populated the British Isles and some of them stayed put. So modern-day Fries and modern-day English are about as closely related as 2 languages can be, but yet they are separate languages. And this sentence in English and in Frisian are pronounced almost essentially the same. Butter bread and green cheese is good English and good Fries. It's a nice little pithy saying. And the pronunciation in both languages is almost identical. But again, the point is that these populations became distinct over time, and that it wasn't just that 2 individuals went and started English and left the population of ancestral speakers on the continent to become Frisians. Okay, so modern genomics indicates not only that we arose through an evolutionary process, but also that we did so as a population. The evidence is such that at no time in our evolutionary history has our ancestral population size been below about 10,000 individuals. Now, this is based on looking at measures of current genetic diversity in humans. So the idea is that we look at present-day humans. We understand processes of recombination, mutation, and that sort of thing. And we can use that to infer in the past what our ancestral population sizes might have been. Now, there's a number of different ways that you can do this, and I go through a number of them in the book. All the methods that we've applied to humans so far agree that we descend from this population of about 10,000 individuals. Now, it's a very common question at that point for people to say, well, where did those 10,000 come from? It's a little bit like asking, well, where did the first English speakers come from? Well, they would have come from a population that was almost identical to them, and so on, and so on, and so on in the past. So these 10,000 individuals don't sort of just drop into existence or pop out of nowhere. We're talking about a population that if we go farther back in time, eventually we would share common ancestors with species like Neanderthals, or I should say Neanderthals now, that's the proper way to pronounce it now. And then species like the Denisovans, Homo erectus, apes in general, chimpanzees, gorillas, and so on. So the idea is that we've had this long lineage that has split along the way, and at no time point in any portion of that whole lineage has our population been below about 10,000 individuals, measuring back at least to about 18 million years ago or more. Okay, so how do we go about doing this? How do we measure population sizes? There's a number of different ways to do this, and it's decidedly tricky to try to explain this sort of material. It's one thing for me to show you a picture of Vitellogenin and pseudogenes in the human genome. It's like, wow, that's very qualitative evidence. I can see it and I can understand it. This evidence is quantitative. It's based on variation, a DNA variation that we see in present-day organisms. So it's a little bit harder. I can't just show you a nice picture of, like, say, a pseudogene or something along those lines. Here's one method of estimating population sizes that employs genetic markers that are linked close together on chromosomes. So if we use just a line to represent a chromosome and we put little hash marks on it to represent different locations on a chromosome, and then we'll use different letters to indicate different DNA letter variations, that are present in those different spots. So those variations could be as small as one single DNA letter difference between you and me, for example. If we look in the present day, and this is just a large population of individuals, this is a pedigree. If anybody's had a genetics class, maybe you've had to deal with some of these pedigrees. So squares just indicate males, circles indicate females, and then the lines just indicate marriages and descent. So if we look in the present day generations, the most recent ones, we might find a number of individuals that have the same genetic markers in the same spatial pattern. Now, there's a couple of different hypotheses for how that might come to be. It could be that mutation events independently mutated to provide those different patterns in the different people. That's not very likely, although it's formally possible. Another possibility is that recombination events that sort of, you might remember crossing over in biology, sort of chromosomes getting together and then breakage and rejoining to mix and match different variation between different chromosomes. It's possible that different recombination events produce that. Again, possible, but not very likely. What's more likely and much more probable is that these individuals have inherited this little snippet of DNA with these different variants from some common ancestor that they share in their past. So they all inherited it from their great-grandpa or something like that, as opposed to independently assembling it. So you can choose markers that are really closely spaced on a chromosome, and the probability of recombination, mixing and matching those is quite low, because the closer things are, the less likely you'll have that crossover event that happens. Or you can also take marker pairs that are farther apart. And if you take those marker pairs that are farther apart, they actually will recombine more quickly, and as a result, they will mix and match more quickly as you go back. So if you take marker pairs that are really close together, it allows us to look a very far distance back in human populations. And if you take marker pairs that are farther apart, then it allows you to look at shorter distances. So one of the nice things about this particular way of doing this is it allows us to scale it relative to time and look back over the last few 100,000 years to see what population sizes may have looked like for humans as we became human. Now, these marker pairs, not that you're going to be able to see that, but what you're seeing there is a phylogeny, or a tree of relatedness, for present-day humans. So these are humans that live, in the present day. At the bottom of the graph would be various African populations. And actually, it's non-African populations that are at the very top. The bulk of this is African populations. You might not know this, but the bulk of DNA variation in modern-day humans is in sub-Saharan Africa. Anybody who's not sub-Saharan Africa comes from the out-of-Africa migration that happened at about 60,000 years ago, plus or minus. And it's a very small population that moves out of Africa and then populates the rest of the planet. So if you're painfully white like me, then that means that you've got less genetic variation than our brothers and sisters in sub-Saharan Africa. Okay, so maybe that's why white guys can't jump, I don't know. So these different patterns of genetic letters, they correlate with what we know about human relatedness. So we see different ethnic groups around the planet that have these little patterns of markers, and they have patterns of markers that are consistent with their ethnic group. So these markers are behaving as we would expect them to behave when we look at DNA sequencing around the planet. Now, this kind of thing is a computational biologist's, they're amazed by this. The ability, this is their fantasy that you can have this amount of data to work with to describe different human populations. So you won't be able to see this, but what this is looking at numbers of marker pairs for different chromosomes in the human genome, and then simply asking the question, based on the diversity of marker pairs that we see in these different populations around the planet, how many ancestors do you need to account for that particular people group? So at the top left there, you've got human chromosome 1. The first column is no, it's not a sub-Saharan African population. It's some non-sub-Saharan African population. They looked at 1.3 million pairs of these markers on the first chromosome alone. They crunched the numbers, and the estimate is that it takes about 2,800 individuals to be ancestors of that particular people group relative to that chromosome. And what you can do is you can just go around the planet and do that for everybody on the planet, all the different people groups, and then just add up the numbers. And when you do, it comes to that about 10,000 individuals for the planet as a whole, for humans as a whole. What's really nice about this particular way of doing things, maybe I'll just go back for a second, looking at different marker pairs like this, is it's a way to estimate population sizes that's independent of mutation frequency. Because these are not new mutations that are happening. This is just the segregation and recombination of already existing mutations. So it's one way to do this analysis without having to make an estimate of mutation frequency. Now, you can do estimates of population size that are based on mutation frequency, and those estimates also come to about 10,000, but they use different methodologies and have independent assumptions. Okay, so that gives us some confidence because we see these different methods of estimating population size that use different assumptions, but yet converge on that same answer. This gives us confidence that what we're looking at is, in fact, accurate. There's another method of estimating ancestral population sizes that actually neither uses mutation frequency, nor does it use estimates of recombination frequency. Now, we can measure mutation frequency in the present, we can measure recombination frequency in the present without trouble, but there are other methods available that actually don't even use those things, and this is one particular example that I like to use. One method of estimating population sizes uses the pattern of relatedness among closely related species. So in some cases, what we will see is certain gene variants that don't nicely fit with the overall species pattern. So I've shown you evidence previously that humans are most closely related to chimps, and then next most closely related to gorillas. Now, one thing to keep in mind is as populations separate and go their separate ways, as they speciate, they have genetic variation within them while they're undergoing that process. Okay? So there's going to be genetic variation present in a population as it's undergoing a speciation event. So what that means is you can have this particular effect take place. This is something called incomplete lineage sorting. I talk about it in the book, and it's a bit challenging to understand, but I'll do my best to walk you through it. If you look at the yellow variant, so here's two different DNA variants, yellow and blue, in this ancestral population before it splits to become the common ancestral population of gorillas and the human-chimp common ancestor, and then later on it also splits. The human-chimp common ancestor population diverges to become present-day humans and present-day chimpanzees. The yellow and blue DNA variants are present in the population of the common ancestral population of all three species before it goes through this process. You can see that the yellow DNA variant is retained in the human lineage, and it's also retained in the gorilla lineage. The blue variant, on the other hand, is lost in the human lineage, and it's lost in the gorilla lineage, but it's retained in the chimpanzee lineage. So what this means is for this particular little bit of DNA, the human genome will actually more closely match the gorilla genome rather than the chimpanzee genome. Okay, so this is called incomplete lineage sorting, and the idea is that not all the variation in a source population will equally sort down. Okay, that's a bit confusing, and it's genetics. Let's take an example, an analogy, and language can help us here again as well. So here I am, a Canadian, speaking to you as Americans. So I don't need to tell you that Canadian and American English do have a few differences, but if you've ever been to the United Kingdom, you would know that the differences between Canadian English and American English are rather small compared to the differences you would experience if you went to the United Kingdom. So Canadian and American English consistently group together to the exclusion of British English, and the reason for that is fairly simple. We share a more recent common ancestral population of speakers than either of us do with the Brits. Okay? So we are closer relatives, as it were, and here's just some automotive terms that nicely make the point. If I came to a gas station in the United States and said that I needed my windscreen cleaned, and you would find the stuff you needed to do that in the boot, and could you please check to make sure I have enough petrol, and so on, that would seem rather odd. So Americans and Canadians group together more frequently than we do with British English. Despite that overall pattern, however, there are certain features of Canadian English that group more closely with British English. One example on that front is the OUR spellings versus OR spellings. So Canadians have this funny habit of spelling certain words wrong, and the Brits do as well. And we do this together to the exclusion of American English. So we spell many words with OUR, and you spell them with OR. Okay, so why is this the case? Well, it turns out that OR versus OUR spellings were actually acceptably variable in pre-colonization Britain, and they were present in the early North American European colonist populations. Again, if you're interested in history, you can look at some of the founding documents of the United States, and they actually have a number of OUR spellings within them. Even though that's like anathema now, it was fine back then. Okay, so what happened here is that the OR forms were lost in Britain and Canada, and the OUR form was retained in those two populations, but lost in America. So the effect is known as incomplete lineage sorting, and not all variants in a source population sort down to every descendant population. So why is this of any use to us in understanding population sizes in the past? Well, if you can find three entities that have incomplete lineage sorting as part of them, then that gives you some information about the source population that led to those three entities. So the source population had both variants present, and also that both of those variants were present in the ancestral populations of the two most closely related entities, either languages or species. Okay. So what this basically does, it allows us to infer that a certain amount of genetic variation in species was present in common ancestral populations in the past. So if we go back to thinking about this as species, it gives us a handle on the genetic variability of those ancestral populations. So if species 1 and species 3 have the yellow, but species 2 has the blue, even though we know species 1 is more closely related to species 2 on an overall level, this gives us information that variants, the yellow and blue variants, were present in their common ancestral population, as well as in the common ancestral population of the source population for all three. So it's a way to look back in time. So what's really interesting about this is that we actually, based on other lines of evidence, had estimates of how often we should see this effect in the gorilla genome prior to our sequencing of the gorilla genome. So we actually were able to predict in advance how often we should see this effect. Now, this effect is dependent on our estimates of ancestral population size, and it's also dependent on having our species related in the right order, which we were quite confident of based on the other lines of evidence that I've shown you. So we predicted we would see this about 25% of the time with the human and gorilla genomes. The actual observation came to about 30%, which is still quite in good keeping with our estimates. And this is dependent on a population size of about 60,000 individuals. So this is now for the gorilla, human gorilla, chimpanzee, common ancestral population. We've actually also done this with the orangutan genome now, which is really quite interesting. So orangutans would be the next group down. We would have parted ways with the orangutan lineage farther back in the past. And as a result, we could expect to see some incomplete lineage sorting between the human genome and the orangutan genome. Because it's been a lot longer since we went our separate ways with orangutan, the lineage leading to orangutans, we actually predicted there would only be about 1.2% incomplete lineage sorting in this case. And we observe about 1%, which again was in very good keeping with predictions prior to that. So this gives us good confidence that our estimates of population size, as well as the time since speciation and so on, are actually in good keeping with what we're observing. So again, we have confidence that the values that we're looking at, because we can predict them in advance, are actually reliable. What's interesting here, though, just to bring the point home again, is that this measure does not depend on mutation, and it does not depend on recombination. We're simply looking at the sorting down of pre-existing variation into present-day populations. This is also why that 10,000 value is thought to be human-specific. So it seems like we didn't dip down to about 10,000 until after we had parted ways with the lineage that leads to present-day chimpanzees. Okay, so these results indicate that humans have precisely the pattern of incomplete lineage sorting that we expect, if indeed we share common ancestral populations with other great apes. So not only is this information about population dynamics over time, this pattern is also very difficult to explain unless these populations are related to one another. This is variation where it's not essential for the functioning of these organisms. Just like it's not essential that you spell it O-R and well, maybe you think it's essential that you spell O-R, but OR versus OUR, Doesn't really matter. Either would do. But we have this particular pattern that we see. So an attempt to sort of explain this pattern from some sort of functional constraint is very, very difficult. Okay, it also shows us that the lineage leading to humans has maintained a large population size over the last 18 million years ago or more. We part ways with the orangutan lineage at about 18 million years ago. So that's why this reaches back to that point. So it also indicates that the population reduction to 10,000 individuals was probably human-specific, as I mentioned before. Okay, so far and so good, but don't we all come from mitochondrial Eve, and don't we all come from Y-chromosome Adam? So you may have heard, it's actually fairly common in Christian circles, to hear that, well, wait a minute, we all come from one woman who lived in Africa about 180,000 years ago. Well, not that Answers in Genesis would say that time frame, but we all came from one woman, and we all came from one man. I think current estimates are Y chromosome, the common ancestor of Y chromosomes is at about 350,000 years ago or so. So how is this the case? Dennis, you're telling me we come from 10,000, but you're also telling us that we all come from one man and one woman? And the answer is yes. Don't worry, I'll explain. So mitochondrial DNA has a particular inheritance pattern. It's only passed on from mother to offspring. So sperm can't transmit a mitochondrion to offspring. So mitochondria are these little subcellular factories that do energy conversion for us. They have their own little circular chromosome, and they're only passed on by the mothers. So next Mother's Day, when you're writing your Mother's Day card, thanks, Mom, for the mitochondria. My wife sometimes says, yeah, that's why I'm so tired all the time. The kids got all my mitochondria. I'm like, no, it doesn't quite work that way, but anyway. I'm tired too, and I didn't pass any on. What can happen in a population? So this is the same population that we looked at previously, the same group. Because mitochondria can get stuck, as it were, they can fail to be passed on if they get into a male lineage. Like, for example, my mitochondria, they're a dead end. They're not going anywhere, right? What that means is you can have certain variants actually take over a population. So if you look at this, we've got these three different colors, red, green, and blue. And you can see that the red variant of mitochondrial DNA actually comes to dominate this population simply because the other variants end up in men or they end up in women who don't pass it on. Similarly, Y chromosome DNA is only passed on through males. So in this example, it's the same group of individuals, but in this population, we have one individual outlined in yellow there, filled in with yellow, who is the common Y chromosome ancestor for this entire population. And the other variants of Y chromosomes have been lost because they couldn't be passed on by females, or they ended up in males that didn't have male offspring, so they didn't get passed down to the present day. Now, regular chromosomal DNA is actually passed on equally by both genders. So there's no barrier to transmission for just regular chromosomal DNA. So what we have here is, in this one group, we have mitochondrial DNA. Everybody's mitochondria comes from that woman at the top right. Everybody's Y chromosome comes from that man highlighted in yellow. But their regular chromosomal DNA comes down from individuals, all of the individuals at the top of this group. So yes, we come from one mitochondrial Eve, who lived about 180,000 years ago, one man who lived about 350,000 years ago, plus or minus, and we come from a population of 10,000 individuals who have passed on their regular chromosomal DNA down to us from the past. Okay, why don't we sum up a bit here? So I've shown you some converging lines of evidence that support the hypothesis that life on Earth is the product of an evolutionary creation, that God used the evolutionary process to bring about biodiversity, including our own species, into being. Moreover, we have converging lines of evidence from different measures that use different assumptions that our species, as it became human, always numbered about 10,000 individuals or more. So, where do we go from here? And this is where my students say, I don't want to be here. I want to come back 150 years from now when it's all worked out, and I don't have to struggle with it. So I would say that a truly Christian approach needs to take both forms of revelation that we have available to us seriously. So we need to take both Scripture and science seriously, neither ignoring one or the other, nor uncritically accepting one or the other. And to that end, don't take what I say for granted. Do your homework. One way to do that is to read the book that I've written with Scott McKnight, or look at other resources. Do your best to inform yourself on these topics and try to understand why it is so many Christian biologists and biologists in general find the lines of evidence for evolution convincing. Okay, but both of these forms of revelation come from God, and we can honor God by doing the best science that we can and also the best exegesis and hermeneutics that we can. We also have to remain humble. We have to recognize that both human science and human exegesis or interpretation are fallible. Now, here's something I'll say that might be a bit provocative, but nonetheless, I'll make the point as well. In cases, however, where we have solid scientific evidence, and that evidence remains strong, perhaps over decades or centuries of examination, Christians are well within their rights, as it were. They have good warrant to re-examine their interpretation of special revelation. And the reason there's twofold. Our exegesis can be wrong. It's been wrong in the past in ways that we have no trouble with now, saying, oh yeah, it's phenomenological language about heliocentrism, no problem. So many of my students just go past that so quickly now. I'm like, do you realize that the effort and time it took to get to that point? And now we are the beneficiaries of that particular process, but it wasn't easy at the time. It won't be easy now, but it's still something that we're called to do as Christians, to look at science and to look at Scripture. And again, if we have the conviction that God is the author of both, then we can be confident that at the end of the day that these will cohere in some way. Okay. I also see myself, my role as a believing scientist in the process is to serve the church by providing the very best scientific information possible. Now, I recognize that others who are also similarly trained don't have the same viewpoints, and that's why I'm glad that we can have a discussion here together today. But I do feel that the church needs to be well served on the science front, such that science can effectively inform our hermeneutics and our exegesis. Okay, I'll point you at a few resources. I've written for the Biologos Foundation for a number of years. I've been with them since 2011 in an official capacity, and there are many resources available on the Biologos website that you could find out more about these topics on. Just a few series that I maybe would highlight to you. I've written a series called Adam, Eve, and Human Population Genetics. I wrote that about a year and a bit ago. And that's a thorough discussion of the evidence, some of which I've shown you today, but also other lines of evidence. And it's also an interaction with a number of Christians who take a different view and my interaction with their arguments. There's also the Evolution Basics series, which is on the Biologos website, biologos.org. And that is intended to sort of be an Evolution 101 sort of series. Perhaps you haven't taken biology in a long time, but you're interested in these topics and you just want to learn what the science of evolution is all about. This is a very large series. It's 50 posts long, and it'll take you from a very simple beginning and then eventually up to a relatively sophisticated understanding of how evolution works. And lastly, of course, the book, Adam and the Genome, that I was privileged to co-author with Scott McKnight. My half of the book is the evidence for evolution and the evidence for population sizes, some of which I've shown you here, but other lines of evidence are in the book. And then Scott says, okay, if this is in fact the case scientifically, what might we think about in terms of re-examining the special revelation side of the equation? Do we have warrant to go back to the Scriptures and say, okay, maybe we've been getting this wrong? And hear me well, science never dictates what our exegesis should be. Science isn't in the driver's seat. What science can do is it can flag up for us that perhaps our exegesis hasn't been accurate. Now, that doesn't mean that science predetermines what our exegesis might find, but it can prompt us to reinvestigate the texts. So science isn't going to determine what we decide exegetically, but it can prompt us to return to the text, just like heliocentrism did in the past. And with that, I'll thank you for your attention, and we'll take our break, I believe. Thank you.
Since it's been a couple hours since you heard from me previously, I'm going to just review real fast, and then we'll dive right into the origin of our own species. We covered the first 3 points in the previous lecture, 3 lectures ago, however long ago that was. Started very broad, asked the question, who do you trust? Because that's really, I think, what it comes down to for anyone who's lay level. And I applied that even to me. If it's a physics question, geology question, that's the same for me. I'm not a geologist, I'm not a physicist. It comes down to who I trust. We also asked the question, why are we still having this debate from the 19th century today? And I argued that it's because this question, the origin of species, of which our own species is a subset of that question is a fundamentally genetic question. And so we have not had the direct scientific evidence with which to answer it until now. That's really the fundamental reason this still exists today. We looked at the non-human species very briefly, both from a genetic and a non-genetic standpoint. I argued that the evolutionary evidences that fail to reject universal common ancestry also fail to reject the modern young earth creationist view. Now I want to focus on very specifically on the human genetic data, and if you've already gotten a copy of the book, you'll know that the chapter 10 on human genetics is organized around these 4 questions. Who do we come from? Ancestry. When did we arise? The time scale. And of course, the big question for this evening, how many people did we come from? And geography is thrown in there too. You heard some of that from Dr. Venema out of Africa. There would be a different geography Ararat, basically, Middle East, for the young earth view. I'll put the population size later. You'll see why in a moment. It's related to the first 2 questions, and actually I'm going to deal with the first 2 together because you can't separate those as well. So this is one of the remarkable facts of the modern era. I'd say maybe 20 years ago you could deal with the age of the earth and divorce it from the question of biology. It's becoming impossible to do so now. Biology is not neutral on the age of the earth. There is a clock in DNA. It records time. And this really presses the question of the age of the earth and biology together. And like I said, it's getting harder and harder to separate them. Now, we basically already covered the question of ancestry in the previous lecture. Patterns of DNA differences, shared DNA differences. Whenever we look in these categories, it becomes very difficult to separate common ancestry or to make any answer to that question of ancestry. Dr. Venema asked a question about pseudogenes, and could we use them to identify baramins? That, I think, gets the right direction. What is the function of DNA? But my answer to that is we know less than 5% of what DNA does based on gold standard genetic tests. I'd say probably less than 1%. If you use the gold standard, humans that have sections of DNA knocked out or disabled, 99% of it we haven't yet tested for function. How many of us would go to Russia if we spoke 1% of the language and tried to say, this is a functional word, this is not? I think it'd be hopeless. That's where we're at genetically. So this is an experiment in progress. The questions of ancestry are yet to be answered. That's an active area of research in young earth creation. And you think about historically, few people have actually had to answer that question. The question of where does ancestry stop and start? The species fixity folks said nothing is related, basically. It was easy. Darwin swung the pendulum the other way. Everything's related. It's only when you are in the middle here, right, basically, that you have to begin to distinguish what are the lines of evidence for common ancestry, what are the lines not. As I implied, basically, in the previous lecture, patterns, I mean, this is a question I'm interested in. I want to know what tools we can use. Patterns don't do a good job. They don't distinguish between ancestry and design. And of course, ancestry and design are part of the young earth model. I'll explain more in a minute. It's really a question of function, and we have very little results of experiments so far that can tell us what's functional and what's not. So really, what we're going to go and where we're going to go, and this is why the time scale is so important, we're going to go to the question of what do the number of DNA differences show? Not the patterns, not the function, but how many differences separate me from you, us from the primates, primates from invertebrates. Those are the questions that we'll now use to try to answer the question of ancestry. And again, this is now very difficult to separate from the question of time. Dr. Venema already alluded to the 2 compartments that we'll be looking at. This is just now to put it in cellular terms. You might be able to see there on the screen there's a 1 and a 2. The 1 designates a subcompartment of the nucleus. 2 is that sort of lavender color. That's the main repository of DNA. That's the nuclear DNA, billions of letters from each parent. And then there's the mitochondrial DNA, the energy factories. And in terms of raw DNA letters, and I'll explain what the letters mean in a moment, a little basic background on genetics. There's less than 17,000 mitochondrial DNA letters in our DNA, comes from mom. There's 3 to 6 billion in our nucleus. So it's 3 billion if you're looking at a sperm or an egg, 6 billion in the rest of our cells. So because the mitochondrial DNA is simpler and inherited in the manner that Dr. Venema described through the mothers, we're going to focus on that first. It also has implications for the nucleus, and we'll see how these 2 compartments interact scientifically and logically. So here's the reason why it's only inherited through moms. Again, as Dr. Venema explained, when sperm meets egg in the oval I just circled, that DNA, billions of letters, fuses with the egg. The mitochondria are towards the sperm tail. Those never penetrate. So that's why it's only through mom. And again, there's just a summary diagram. Nuclear DNA from both parents, mitochondria only through the maternal line. Now, I'm going to use two analogies by and large for what DNA is, just so we're all on the same page. This is the diagram of the structure I showed in the previous lecture. It's this twisted ladder-like structure, so there's the analogy number one. It's like a twisted ladder, and we'll explore that a little bit further in a moment. I've also got letters assigned to it, and you might be able to see the different colors for those rungs of the ladder. So the second analogy is that DNA is like a language, and Dr. Venema explored that analogy at length. It has an alphabet. It's a chemical alphabet, and there are only 4 letters in that alphabet, A, T, G, and C. So this is the structure that gets passed on in sperm and egg. It has to be copied first into sperm and egg. That copying process is imperfect. So you can think of it, they're mutations. You can think of mutations as basically a color change in this diagram to the rungs of these ladders. Mistakes happen, copying errors happen, and because they happen, we then can compare 2 DNA sequences next to one another. So you can see in this diagram, there's 20 rungs, 20 letters that I've compared. 4 are mismatches due to the mutations that I animated. So we would say there's 80% difference between these 2 sequences, 80% identity between these 2 sequences, 20% difference. You might say, well, how can you know you're lining up the right thing? Well, for humans, as an example, we share so much DNA, it's easy to line up, match, 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 and every now and then a mismatch. So you can be fairly confident you're lining it up the right way. So the question is, what do the number of these DNA differences show? We can take the DNA from every one of you in this room, line it up, and get a number. And this has been done now for all sorts of people groups around the planet. We have over 10,000 nuclear DNA sequences. We have over 30,000 mitochondrial DNA sequences in our public databases from individuals around the planet. So what do these differences show? We also have mitochondrial DNA sequences from over 6,000 animal species. And over 800 mammal species. So there's all sorts of comparative analyses we can do. You can find nested hierarchies in mitochondrial DNA sequences, but once again, that fact, that pattern, doesn't distinguish between ancestry and design. So what about the number of DNA differences? This is where the time scale comes in. So evolution would say that all DNA differences ultimately are the result of mutation, this process of mistakes. And that the number of mutations in between any 2 species, particularly at the mitochondrial DNA level, is a product of the time scale since they last shared a common ancestor. So humans and chimpanzees, and I've shown an African woman here just because evolutionists would say the Africans evolved first. Traditionally, they put the time scale at 6 million years ago. Some would say 13 million, and I'll explain that more once we get to nuclear DNA. I'm just going to calculate it for the whole range. There are textbook equations for predicting the number of DNA differences over a certain time scale when you're dealing with mitochondrial DNA in 2 different species. It's this equation, and this is really all the math you need to know for this lecture. So the number of DNA differences is a product of the mutation rate times the time and times 2, because mutations are happening in each lineage, and we count the differences by comparing the 2 to each other. We know the mutation rate in humans is about 1 every 5 to 8 generations. It's been the subject of over 15 studies, highly contentious. Also highly divergent in the amount of statistics each study weighted their analysis with. So anyway, you distill all that away, it's about 1 mutation every 5 to 8 generations. We don't know the rate in chimpanzees, so for a starting point, we'll just assume it's the same. When we look at nuclear DNA, we'll see that this is a pretty fair assumption. So 1 mutation every 5 to 8 generations. So think of it practically. Again, I have my mother's mitochondrial DNA, my kids have my wife's. You'd have to go back 1, 2, 3, 4, 5, 6, 7, 8 generations before you found a difference in my lineage. What would happen in 6 million years of this process in human females and our female ancestors and the chimpanzee female ancestors? Well, you can put numbers on this. And at a minimum, and actually let me back up and make one more technical point I just realized. Mutations per generation, but we're going to multiply that by years, so if you're thinking in math terms, the units don't line up. You have to convert mutations per generation to mutations per year. And the way you do that is by knowing something or assuming something about the ages at which females reproduced. Some women give birth in their teens, some women very late, maybe even 50 years old. So what we're going to do then to compensate for that range is calculate and make predictions over that entire range. And these are the results. So if all women were ages 50 in the human lineage and all chimpanzee ancestors were ages 50 in the chimpanzee lineage over the past 6 million years, there would be a minimum of 28,000 differences that are predicted in between these two species. Now, I mentioned earlier the mitochondrial DNA sequence is only 16,000, 17,000 letters long. How can that be possible? What that means is you would have mutated position 1, and then 2 at some point, and 3, and all the way down to 16,569, and then you do it again. And again, that's practically what it means. If all women were ages 15 over the last 6 million years, 13 million years, you'd have over 350,000 DNA differences. That's the top of that black line on average, if they're ages 25 or so. 150,000 to 200,000 differences. So again, mutating over and over and over again, that's what it means. And you can see there on the right, the actual number of differences between our two species is only 1,400. And we'll talk about some potential explanations for this in a minute. What I want to do first is look at other evolutionary time points in our lineage. So evolutionists would say we split from Neanderthals about 400,000 to 700,000 years ago. Again, Africans evolving first in the modern human lineage. You could use a similar equation for this split. I'm going to treat the Neanderthals and humans as the same species, so you drop the 2. For technical reasons I won't get into. We can add the 2 back, but I'm trying to be generous here, and you'll see why in a moment. So how many differences should accumulate between humans and Neanderthals in 400,000 to 700,000 years? We'll again use the human mutation rate, 1 mutation rate every 5 to 8 generations. Convert that to years, depending on how old women were when they gave birth. So if all women the last 400,000, 700,000 years were 50 years old when they gave birth, nearly 1,000 DNA differences would have accumulated between Neanderthals and humans. That's the bottom of that black line. If they're all ages 15, nearly 10,000, and then on average 25, then more like 5,000. And today there's only 213 differences. So again, another overprediction, not as dramatic. We're not mutating every single letter, not every single rung of the DNA ladder, mitochondrial DNA ladder. One more time point, and then we'll talk about potential explanations. 200,000 years ago is the evolutionary origin for the modern human species, and as Dr. Venema alluded to, 50,000-ish, 75,000-ish years ago, non-African people groups split off from the African people groups. We're all part of the same species. We use this same equation that we used for Neanderthals. How many differences would we see? If all women were ages 50 over the last 200,000 years when they gave birth to those ancestors in our lineage, 477 at a minimum, bottom of that black line. Over 2,000, nearly 2,500, 3,000 if they're all ages 15. You can see an average about 1,500. In Africans, there's only about 80 differences on average. Non-Africans, about 40. And we'll talk about reasons for the differences between these two people groups in a moment. So what could be some potential explanations for this discrepancy? And these will become important, especially when we start discussing the nuclear DNA sequences. Could it be that we need to look at different levels of ancestry? Maybe we're too focused on human chimp or human Neanderthal. What if we go back further in time? We'll end up putting more differences in the actual column, let's say humans and orangutans, humans and a rhesus monkey. But we'll also add to the predicted differences, so that wouldn't solve the problem. What about the time scale? Now this is really interesting for 2 reasons. Actually 3. The third one will arise when we discuss the nuclear DNA. What happens if you change the time scale? Well, the evolutionary time scale is interconnected, so you're going to have to begin to rewrite significant sections of the evolutionary narrative. Second reason, or maybe now up to 3, third reason this is significant, is thinking more deeply about how the evolutionary time scale, specifically in geology and astronomy, is established. It's established on the assumption of fairly constant rates of change. Why do I say that? Because young Earth creation astronomers and geologists have questioned that assumption for many years. They'd say in geology, as an example, that Noah's flood, a global flood, would dramatically alter rates of erosion, deposition, so forth, that the rates we see in the present would not be a good indicator of the rates in times past. Well, now I've taken that assumption to genetics. I've assumed constant rates of change. And it gives you the opposite conclusion. So that begins to create some logical dilemmas there as well. Now, the third explanation, which is probably the most likely, is, well, what about natural selection? Could natural selection play a role? Plausibly, the answer is yes. Mitochondrial DNA encodes for some of the key proteins in that function in mitochondria. Now, your heart is highly dependent on mitochondria. You're sitting here breathing, listening to me because you have functional mitochondria. So what would happen if your mitochondria got hit with a whole bunch of mutations? You might keel over and die. So that would seem a priori to be a fairly decent explanation. So we'll go with that for the moment, and we'll raise that question again when we look at DNA. But there's another question we need to ask, and I'm going to go back to Dennis Venema's book again about how you define a hypothesis and a theory. One of the defining features of a good theory is that it makes accurate predictions about the natural world. And this isn't just Dr. Venema. He's here today. We're discussing with one another. I'm using this, but you can go to evolutionary textbooks. You can look at the history of the creation-evolution debate. This is primarily the major arena of criticisms of the young Earth model, or the creationist model in general. Evolutionists have said for years, you creationists say God did it. How does that advance science? What testable predictions does that make? How does it advance our understanding of the natural world? And you can see, if you've ever done any sort of science, or you can just believe me when I say it, if you haven't. Let's say you're doing an experiment. I'm trying to understand how cancer works. I've got this drug that I think is a potential candidate. I'm going to test it in the lab. It doesn't work. Oh, God did it. How does that advance science? How does that lead us to the cure? You can see why that sort of idea would be a strong challenge to the creationist view, at least on the surface. My point in bringing it up here is the sword cuts both ways. Let's say natural selection is the explanation for the discrepancy. What testable predictions does that make? For example, one of the most divergent people groups on the planet, most mitochondrial DNA differences, are the Khoisan peoples, or the Click speakers of Southwest Africa. No one has measured the mutation rate in the Khoisan peoples. What testable predictions about the mutation rate in these peoples does the hypothesis of natural selection make? That's really the challenge, not even from my own lips. That's the challenge the evolutionary community has to meet because they set it. They set that standard. And it's a good standard. It arises just out of the nature of science. Well, let me throw out another hypothesis that could explain these data. Of course, I'm going to go here because I'm a young Earth creationist. We would say all peoples go back to Adam and Eve. So we'd say in the beginning, Adam and Eve both had mitochondrial DNA. Adam is a man. He doesn't pass it on, so we go back to a mitochondrial Eve who lived 6,000 years ago. So the differences we see today should be a product of the mutation rate, one mutation rate every 5 to 8 generations, times 6,000 years. And when you do this prediction, if all women were ages 50 over the last 6,000 years, there would be around 15 differences in the global human population today. If all women were ages 15, there'd be around 80 differences. And you can see that this range of predictions, the top and bottom of that black bar, captures what we see today. About 80 differences in African people groups, about 40 in non-Africans. You can say, okay, the non-Africans, that fits maybe an age of about 25 years on average, what we kind of see today. Isn't that straining things to say that all African women would have been ages 15? Yes, and maybe not. Why? Because we have data from the United Nations. Let's say the data from 1976. Marriage data is not the same thing as age of reproduction, but there's probably going to be a correlation. So 1976 data, how old were women married? And the data now is in terms of nations, African nations, non-African nations. If you look at, let's say, ages 30 to 34, the column sort of near the middle of this, you can see the percentage of African women who were married. It was around 80%. The percentage of non-African women, about 77%. Hardly any difference. If you look at the early ages, so the top row, first column, there on the left, how many African women were married at ages 15 and 19? 33%, nearly a third, exactly a third. How many non-African women? Only about 10%. That's about a 2 to 3-fold difference. That's about a 2-fold difference right there. Now, that's data from 1976. That doesn't mean this has been historically true. But you're beginning to see how there might be another explanation for why there's various differences between Africans and non-Africans that have nothing to do with who originated first, but rather people group specific differences. I'll explore that question and return to it later. I want to show you this tree, though. This is several hundred human individuals. I think Dr. Venema showed you a tree also. The tips of these branches represent individuals. Way off to the right is where you'd find some of the Khoisan peoples. They have very long branches. Now, the length of each line, there are really strong parallels to a family tree here. So if you look at a family tree, the amount of difference The distance between two peoples would be the number of generations. This is not a display of generations per se, but the number of mitochondrial DNA differences. So if you have a very long line, a lot of DNA differences. If you have a short line, hardly any. And the way you find the difference between any two people is you find the tip, let's say, at one of these Khoisan peoples, and you trace that line back to where it connects to another person, and that tells you the amount of difference. There are some things I want you to notice about this tree. So this is from various people groups around the globe. Notice how so many of these lines come together into 3 nodes, and that's not an artifact. This is a fact the evolutionary community recognizes. They call them haplogroups. This is the L, M, and N haplogroups. The second thing I want you to notice, not only is there 3 major groupings, you might see, well, there's some subgroupings. There are subgroupings, and we'll talk about them in a moment. I want you to notice the length of the lines that connect these 3 major groups. I've highlighted them here in blue. They're fairly short. So the lines connecting the groups are short. The lines radiating out from these groups are long, and we can draw an even longer one for the Africans, or I guess that direction. Let's think about this tree then from a young earth perspective. What would be the maternal ancestry from our reading of Scripture? It would go back to Eve, population would grow, shrink to 8 at the time of the flood, regrow again, briefly, split at the Tower of Babel into multiple people groups, and then the rest is history. So, there were 2, actually you could probably say 3 major bottlenecks. So you have creation, Adam and Eve, then you have 8 people at the flood, and then the different groups at the time of the Tower of Babel. So you got short branches among the 3 major groups, longer branches radiating out. Let's think in terms of time now. You have from Genesis 5, you take those time stamps plainly for the paternal lineage. You've got about 1,700 years between Adam and the flood, but only 10 generations. They lived long. And then you see, after the flood, roughly, we'd say probably 4,200, 4,500 years. So that's about a 3-to-1 ratio almost between post-flood time and pre-flood time. And the difference gets bigger once you think in terms of generations. It'd probably be fair to say that maternal ancestry was also long pre-flood. Maternal lifespan, just like paternal lifespan in Genesis 5, would have been long pre-flood, shorter post-flood. So you'd predict that pre-flood mutations would have few opportunities, few generations. Post-flood, there have been much more generations. So what are these 3 nodes? I just gave away the answer, and I meant to build up to the punchline in a minute. So there's Eve in the beginning, 1 Eve. Then the ancestry shrinks to what? You have 8 people, Noah, Mrs. Noah, 3 boys and their wives. 3 boys get their mitochondrial DNA from Mrs. Noah. And then it stops. Genesis 9, from these 3, the whole world was repopulated. We don't get our mitochondrial DNA from them, we get it from their 3 wives. So the fact that this then breaks into 3 with relative branch lengths that fit the relative time proportions seems to fit this biblical model. Short pre-flood times, those blue bars separating the 3 wives. Scripture doesn't say if they were sisters or more distantly related. And then lots of generations post-flood, those pink bars. So the structure and relative branch proportions and the absolute numbers, 3 independent lines of evidence, all fit what we see in mitochondrial DNA. Well, what about these subnodes? This model makes testable predictions. If we're all coming from those 3 nodes, and DNA differences are a marker of time, what we're looking at then when you radiate out from those nodes is post-flood time. In other words, the history of civilization. This model predicts you should be able to read the history of civilization off this tree. The history of the Roman Empire, the history of the Persian Empire, the history of all Greeks should be here. So what about this subnode? You see that it's coming off one of the major 3 nodes, and about halfway up it begins branching. Where do these, so the tips of these branches are individuals. Where do these individuals exist today? So let's put this together. I just hit the wrong button. Back up 1. And I didn't want to hit that, so let me go back this way. There we go. So you can see in terms of relative time, there's a branch that goes up from that node, then about halfway up that branch it splits off. So what would be half of 4,200, 4,500? It's 2,000-ish, 2,300 years ago. What happened 2,000-ish, 2,300 years ago around this section of the globe? The Roman Empire covered this section of the globe. So this is just the beginning of the sorts of studies we're doing. And in fact, if we had time, I could tell you some really electrifying results that we've been doing with the paternal ancestry, the Y chromosome. Rob Carter and I have been doing this, Creation Ministries International. We've been able to date the transatlantic slave trade. We're looking at the signature of colonialism. This is really working. It's really incredible. And that's not the only testable prediction it makes. Let's go back to the Khoisan peoples. Where are they? Way out there. I said the average number of differences is about 80 in Africans. The mutation rate has yet to be measured in some of these most divergent African people groups. The mutation rate I'm using has been measured in non-African people groups. So I would predict then, and this is a testable prediction, a falsifiable prediction, that the mutation rate will be 1 to 5 to 8 generations, perhaps faster, 1 every 2.5 to 4. So the prediction will be 1 every 2.5 to 8 generations. You can test that in the lab as soon as some Khoisan people give us their DNA. And these results, we're focusing on humans. These results are true of the other species in which we have measured mitochondrial DNA mutation rates. It's in the book chapter. I've got published papers on this. We don't have time to go into it. So why am I only showing 4 other species? You think of the history of genetics, it's only recently that we've gotten DNA sequences. It's even more challenging to get the mutation rates, so that's why there's attrition. The number of species with DNA sequences is a whole lot more than the number of mutation rates. This is the data that we got, and it's all pointing towards a 6,000-year time scale and challenges a millions of years time scale. So that's mitochondrial DNA. What about the nuclear DNA? Let's once again make testable predictions. The question is, what are the number of DNA differences? Patterns don't tell us much. Function is still an experiment in progress that'll take a long time to figure out. What do the number of DNA differences show? What do they tell us about our ancestry? And again, let's go with the evolutionary model first. Evolutionists would say about 6 million years ago, 4.5 to 6 million years ago, that's traditionally been the answer. Human lineage and chimpanzee lineage split, African peoples first, which is why I'm showing the African lady. Biologos website has used the number 6 million. You can see it here, genetic evidence for evolution. So let's go with 6 million first, and there's a reason why I'm going to do this. We'll use a similar equation. That's actually the equation that the authors of the chimpanzee mutation rate paper used. So in this case now, nuclear DNA, we have the human mutation rate, which has been measured numerous times in the nucleus. That happened first, 2014. The chimpanzee mutation rate has been measured. They use this equation. How many differences should exist between humans and chimpanzees? It predicts about half of what we see. Now, let me do a segue here. Dr. Venema mentioned Jeff Tompkins. We're comparing 2.2 billion of our total letters. There's total number of letters in humans and chimpanzees is over 3 billion. So where we align, so they're looking at a particular type of DNA change, SNPs, single nucleotide polymorphisms is the technical term. There are other sections, you can read in Dr. Venema's book how you get to 95%. Well, there are also whole chunks that are not found in either species. You can see that in the original chimpanzee genome paper. You can get a number of about 89% identical if you include the single letter differences, the chunk differences, and the big chunks that are missing in either case. That's been Jeff Tompkins' point all along. You see in the chimpanzee paper, 89% only matches up. You can find a match. You see in the bonobo paper, so sort of another chimpanzee species. You see it in the gorilla paper. You see it on the Ensemble website, one of the public repositories of nuclear information. There's only 89% that can find some sort of match. In that 89%, you can find 95, 98% identity. But the overall number of differences is upwards of 300 million if you include everything. So there's a big gap. If we focus on those to which only those sections to which this mutation rate applies, as the authors of the chimpanzee genome paper point out, 6 million differences predicts only half of what we see. So if we say, let's just focus on that 1% difference, you can only get half of that by mutation. So what's the explanation? We're not going to change ancestry. That'll run into problems. What about time scale? Well, if you bump the time scale back to 11 to 17 million years, around 13, 14 million years, you exactly capture the difference that we see. That's the option that the authors of the chimpanzee genome paper published. That was their conclusion. We should bump back the time scale. You can read it for yourself, Science Magazine, 2014. Venn is the first author. What questions does that raise? The evolutionary time scale is interconnected. So if you're going to bump back one part of the time scale, well, you're going to bump back the chimp-human lineage split from the gorilla split, and then the gorilla-chimp-human split from the orangutan split, and the great apes from the other monkeys. Or from monkeys, I should say. So chimpanzee human, if we bump that back from 6 million to 14 million, chimpanzee gorilla from 6 to 8 million to 21 million, gorilla orangutan from 12 to 16 million to 40 million, You're going to have to bump back the rhesus macaque orangutan split to 74 million years ago. Why is that significant? The evolutionary timescale puts the extinction of the dinosaurs at 65 million years ago. The first primate fossils are found around 55 million years ago. You're pushing back even this split beyond the first primate fossils. So you can see what happens when you start to rewrite the evolutionary timescale. That's what the authors went with. That's not my conclusion, that's theirs. Now, natural selection. Why am I bringing this up again? Well, let's go back to mitochondrial DNA, because these two compartments go together, nucleus and mitochondria. So the evolutionists, the authors of the chimpanzee paper are saying, let's bump the timescale back 13 million years. That means you're going to have to do the same thing in mitochondrial DNA. The minimum number of differences is not 28,000, it's now 61, 62,000. Well, natural selection can eliminate 28,000, it can eliminate 62,000. Okay, natural selection here. What about here? What about in the nucleus? What role does natural selection play? By definition, it cannot play any role in this question, because the original prediction was an underestimate. If natural selection functions to eliminate differences in mitochondria, you cannot eliminate any more differences in nucleus without making this discrepancy worse. So that raises the question, when does natural selection apply, when does it not? And I can even put it in the language of the evolutionists from 50 years ago. They say God did it, stifles science. Natural selection did it. Is that really what we're looking at now? Is that the state of evolutionary science? What creationism was accused of being 50 years ago? There's actually a fourth explanation, and I want to illustrate it from the evolutionary explanation for the origin of modern humans. So we could say, okay, our species, the modern human species, originated 200,000 years ago by the evolutionary time scale. We'd use a similar equation. Now, technically, this equation is a function of population size. I'll show you why I'm ignoring it for the moment, because it's going to be an underestimate. I'll show you in a minute. We'll use this equation to predict how many mutational differences should arise over 200,000 years, and it's only about 500,000. And today, in African peoples, in our nuclear DNA, there's about 4.3 million letters difference. In non-Africans, it's less than that, 3 to 4 million. And you only predict about 10, 12% of that. So is this an erroneous prediction? I said earlier, does evolution explain all differences by mutation? Ultimately, yes. Each generation, no. Why would I say that? Well, let's go back to this diagram and the modes of inheritance. So mitochondrial DNA is maternal. Nuclear DNA is from both parents. Now, none of us in this room are going to have parents who are clones of each other. In fact, probably everyone in this room does not have parents who are even close relatives. So your parents will have different DNA sequences. They will each pass on a DNA sequence to offspring. So that means this offspring is going to look different genetically than the parents. What percentage of those differences are due to mutations each generation? Less than 0.1%. Most of the differences you have within your body, and I'll explain what I mean more in a moment, are due to inheritance, the pre-existing variety. So that's what the evolutionists would invoke here, that our species originated 200,000 years ago, but we've been mutating back to the split with the chimpanzee lineage, and so those from, 6 million, 13 million years ago to then, those mutations were accumulating, and it's going to be a function of population size, but then those were inherited, and the vast majority of the differences between me and you are due to that and not to mutations in the last 200,000 years. So they'd say inheritance. So that raises also the question, when does that apply, when does it not? Could we apply it to the human-chimpanzee question, the chimpanzee-gorilla question? Well, now let's get to the creationist model, and this will get us then closer to the Adam and Eve question. But you see why we have to cover these questions first. And hopefully you're also able to see that the questions of ancestry and time are interlinked. Biology is not neutral on the time scale. You have to deal with it. So in 6,000 years, obviously you're going to explain just a tiny, tiny fraction of what we see today. But if evolutionists invoke inherited differences, why can't I? Now you might say, inherited from what? You're not invoking 6 million years of ancestry. So let me answer the question by asking you a question and see if you can answer it. You don't have to say it out loud. So I'm going to speak now to those who maybe would hold to a literal Adam and Eve. You read Genesis 1, God says, be fruitful and multiply. Do you think he intended for that command to be fulfilled by cloning? Well, what a weird question. Why would you ask that? Well, if you think that's weird and you'd say, no, what a strange answer. No, I don't think that's what he meant. You've basically acknowledged a particular genetic view without realizing it. So this is just a little bit more genetic background. So I've said DNA is like that twisted ladder-like structure. You can see that in the lower left-hand corner. It's also like a language, A, T, G, and C. You can see those color-coded there. Well, DNA doesn't exist in this billion rung long ladder that just kind of sticks out the end of a cell and pokes through. It's wound up and gets wound up and very tightly when cells divide, and it forms these X-like structures that are chromosomes. And this is a real diagram, picture of human chromosomes. And I want you to notice something here, and this will explain more what I was saying about 99.9% of our differences being inherited. Notice that there are some large chromosomes. Now, these are real chromosomes, so they weren't nicely electronically lined up. They're kind of floppy. These, noodles is what you can think of them as. Wet noodles. And there's some small chromosomes. But notice that for every long chromosome, there's a corresponding pair, and for every small one, there's another corresponding small one. So the fact here that I want you to see is that chromosomes come in pairs. And in our DNA, there's 23 pairs of chromosomes, 46 in total. Now, you might notice in number 23 that there's a long paired with a short. That's the sex chromosomes. This is an XY individual. Y is short, X is long. This is a male. Females would be XX. Now, let's just focus on chromosome 1. It'll help us understand what's going on here in the biparental mode of inheritance. So we have pairs of chromosomes because one member of each pair comes from each parent. So you can diagram it, crudely this way. Dad contributes one member of each of the 23 pairs. Mom contributes one other member. Now, it's not like Dad contributes the information for the right arm and Mom for the left. They both contribute for the entire body, and then in a way we still don't fully understand, but it was something that Mendel observed. They interact, and your unique characteristics come out of that. So, back to the question, did God create Adam and Eve to reproduce by cloning? If your answer is yes, that means Adam had DNA sequences like this. He wouldn't have had just 23 individual chromosomes, not 23 pairs. That's lethal. So he would have had pairs like this. If you say Adam and Eve reproduced by cloning, he looks like this. If your answer is no, which is my guess is what most of you would say, that means you're proposing Adam was created with differences, or Adam and Eve were created with differences between one another. You're basically saying there were pre-existing differences created into Adam and Eve. If you say, no, they did not reproduce by cloning. That's where these pre-existing differences would come from. So, mutations contribute very little. Inherited differences contribute a lot. This is simply using a page from the Evolutionary playbook in terms of the last 200,000 years into their model. I'm just putting a different number on it. What would be the signature of this? Scientific ideas make testable predictions. Well, here's, once again, the model of population growth that I described earlier. Adam and Eve, 2 people, then they grow massively. Earth gets filled with sinful people who apparently were violent, so God decides to wipe them all out at the time of the flood. Population shrinks to 8. They grow again quickly. You look at how many children each of the patriarchs had in Genesis 10. It's massive population growth, splits at Babel, and then the rest is history. Now, let me summarize a lot of population genetic theory here. In a moment. I'll use a simple illustration. So the average rate of nuclear mutations between generations is about 80. It depends on the father's age and so forth, but on average about 80. So that means there's about 80 mutations that my son has that neither me and my wife have. Now, what's the chance that his 80 rungs, 80 letter differences are going to spread around to the 7 billion people on this planet? It's virtually zero. So population genetics theory is, when the population size is large, new mutations are statistically rare. So if this is your population model, the signature of this event would be a large number of rare differences. Evolutionists acknowledge this. You can look at the evolutionary literature. They say there's an excess of rare variants in our population due to recent explosive population growth. You can actually predict this. The number of rare differences is quantifiable in our population, and it fits exactly what you see after 6,000 years. This is the signature of the creation flood bottlenecks. There's another signature. So if this was Adam's DNA, 2 different versions, even though he had no human parents, God creates him with the potential for diversity. This gets recombined. Chunks of the chromosomes get swapped. There's actually 2 processes. Recombination swaps big chunks. There's another process called gene conversion that sometimes just swaps single letters. So these original chromosomes will get all mixed up over time. And you can see these chunks. Scientists would refer to these as haplotype blocks. That's the technical term. You can predict how many haplotype blocks you should see in the human population today, and you see them. you see exactly what you'd expect to see from the young Earth model. Now, let me stop here for a moment. Most of what I've shown you is actually not a prediction. It's a retrodiction, taking what we see today and saying, does it fit this model, yes or no? Prediction is saying things for the future that have yet to be investigated. So I gave you some of those for mitochondrial DNA, the Khoisan people mutation rate, reading the history of civilization off mitochondrial DNA. You should be able to do the same thing here. It's a little bit more complicated because you're dealing with a mutational process and a pre-existing inheritance process. If you're interested, I can tell you how. I'll be giving a complicated diagram in my book this fall that shows how this could give you a relative order of events. So that's the first two questions, ancestry and timescale. And I've given you some arguments for a recent origin, and I've shown you things that are consistent with coming from Adam and Eve. Now, Dr. Venema says in his book, and this is not to be contentious, but this is a dialogue. He says, to date, every genetic analysis estimating ancestral population size has agreed that we're descended from a population of thousands, not single. Now, Rob Carter has published the preliminary data that I've just shown you here as much as 6 years ago. He's published a number of papers modeling biblical population growth, very detailed simulations of population growth and crash. The data I just showed you I published last April, a year ago. So these data that I just showed you are found in that paper. So I would say it's incorrect to say that every genetic analysis agrees that we're coming from 1,000. What about the claim that many of these methods are independent of one another? That's in one sense true, but there are also some assumptions that are common to all the methods, the 3 lines of evidence that Dr. Venema showed you that are also in this book. 1, assuming that mutations are the source of all variety, and 2, they assume a long time scale, and the third one, of course, assumes common ancestry. So one of the lines of evidence that I think he included in the book and has been published elsewhere is that there are too many differences to explain from 2 people over 6,000 years. That's true only if mutations are the source of all DNA differences, if you include pre-existing differences. Basically, if you say that no, God did not create Adam and Eve to reproduce by cloning, this argument goes away. He also made an argument from linkage disequilibrium, though I don't think he used that technical term, and we're all happier for it, because and he makes a funny story in his book about why scientists come up with these crazy terms. So I'm going to spare you the math details and just give you the principle. It comes from basic population genetics. And pretty much all the arguments for or against an ancestral population size are statistical in nature. This is how it works. I'm going to use a silly example just to illustrate the point. Let's say you reach into a jar of jelly beans and pulling a handful out or a certain number out. I'm going to use this to simulate the process of reproduction. You might think that these jelly beans represent individuals in a population. Instead, they represent sperm and egg. So this is weird, but population genetics is weird. Sorry. So what would a bottleneck look like? You think, well, you just take one sample out and then you take 2, and you can do statistics for this. And what's the chance that both are going to be pink or both are going to be black? So in this case, sperm and egg, because we're dealing with nuclear DNA, you actually have to pull out 4 jelly beans. There's a chance, there's a chance that all 4 are pink. And I'm oversimplifying this. Normally, discussions deal with multiple generations, but the screen is only so long, so I'm going to deal with 1. There's a chance, statistically, that these are all pink. More likely, there's a chance that you'll have a not 50-50 split, and that over time, if you keep the population size small, statistically then, you'll lose variance, and eventually the population will be fairly homogeneous. Now, I'm using this as an example of one particular DNA position. You could do this for, there's millions of differences among us, so you'd have to do it for millions of colors, but that gets messy too. And it doesn't matter which color you pick, the principle is the same. You pick out a few. Statistically then, over time, your population has a strong probability of being homogeneous. And let me contrast then to a non-bottleneck, basically. So if you take a handful of jelly beans, all of them, dump them out, and you say they reproduce themselves, and this is going to be the basis for another population, well, you're almost guaranteed to not lose anything. It's a statistical argument. Again, in contrast to a bottleneck, you're going to become fairly homogeneous. So how do calculations of ancestral population size work? We're basically starting like this. We don't have the history. We just have what we see today. Is there a lot of differences, or are there a few differences? Does it look like we went through a bottleneck? Does it look like we've always been this handful, big handful, the whole jar? And that's really essentially the various lines of evidence that Dr. Venema has presented. This is basically what he's saying. We have a lot of differences. It doesn't look like we passed through a bottleneck. It fails to reject the hypothesis of a population. It does reject the hypothesis of this particular bottleneck. Well, it also fails to reject the hypothesis that God created Adam and Eve with created differences and that they grew exponentially. What happens if you have created DNA differences? And let me back up here. Again, I've shown 4 because Adam and Eve each have 2 copies. So they're going to reproduce, but they're going to reproduce a lot, be fruitful and multiply. So they're going to produce a large population of individuals, virtually guaranteeing that nothing is lost. And in technical terms, the math equations you'd use to calculate population size with the exponential population growth change. So the evolutionary literature fails to reject this hypothesis. It's essentially a straw man to say that these lines of evidence reject it. And then, of course, the last line of evidence, independent lineage sorting, assumes human primate common ancestry. So if you assume the evolutionary ancestry, then you can make calculations that argue for population size. So I would argue, in contrast to what he claims in the book, that every genetic analysis rejects Adam and Eve only if you exclude certain explanations and hypotheses from the start. He also claims in his book, anti-evolutionary scholars have not yet mounted a convincing response to population genetics evidence, nor is it clear that they will be able to do so, since there does not appear to be anyone in the anti-evolutionary camp at present with the necessary training to properly understand the evidence, much less to offer a compelling case against it. I'll let that stand as it is. So here's the 4 questions that we've discussed, that I discussed in the book chapter. On the question of ancestry, the major arena in which the predictions will be tested is in the arena of function. Patterns do not distinguish between the various models. Function is the arena, and unfortunately we know less than, probably only about 1%. We've only done experiments on 1% of our DNA in terms of gold standard. Timescale, hopefully what you've seen in this hour, is that the young Earth model makes testable, falsifiable predictions that we can go into the lab today, tomorrow, and explore to see if they're true. For the people groups, and in fact, again, if we have more time, this hour is focused on humans, I can make predictions now for species around the planet. This isn't just limited to the origin of the human species. You've seen that the young Earth model from Adam and Eve makes testable predictions. I've made retrodictions mainly, but you can see there's predictions as well, reading the history off of our DNA sequence. And again, if we had time to look at Y chromosome, I think there's really some electrifying results that are showing up there. Hopefully what you've also seen is an indirect answer to the question of geography. Why are there differences between Africans and non-Africans? Why is there more diversity in Africans than non-Africans? So I've shown you marriage data that will affect mutation rates. What I didn't show you is a paper from 2011 looking at recombination rates in African Americans. And what they found is that the rate is faster. And the authors actually concluded in that paper saying, and I could show you the quote, that, maybe there's more to the African differences than just different size population bottlenecks out of Africa. They basically conceded there's genetic differences between these populations that may have nothing to do with their history. So I would say the argument that the evidence is consistent with a simultaneous origin of the major ethno-linguistic groups consistent with the Tower of Babel. Now Scott McKnight says on the Biologos website, when he was they ran a series promoting the book. He said, Scott McKnight says, I'll put this stronger. If you don't accept Dennis Venema's section, then my section of the book need not be read. So basically, he says, we're reading scripture after genomic science. Let's flip the argument around. In light of what I've just shown you, what can we say about Paul's arguments in Romans 5? What can we say about Paul's arguments in 1 Corinthians 15? I think it sheds new light on the subject. What's the significance of what I've just shown you? Once again, I think we've come a long way from the 1950s. The young Earth model has matured significantly. And this is, to me, just a function of time. Many, again, would point to the publication of the Genesis flood, 1961, as the beginning of the modern young Earth creationist revival. And at the beginning, you're starting it, so you're going to have few people to go along with you, and you're also taking on a massive scientific enterprise. So the posture the early young Earth creationists took was largely defensive. It's just the natural consequences. Evolutionists would say this, and young Earth would say, well, not so fast. Well, what about this? And here are some problems. Fast forward to today, I would be a second generation, if not a third generation young Earth creationist. We've got a large community of scientists who work on things together. On top of that, we finally got access to the genetic data, the most important scientific data in this debate, and it is freely available. So people you can sit at your laptop, if you know the right tools, and do your own analysis, because all this data is out there. So you've got a bigger community, a whole lot more data that's freely available. This evens the playing field and allows young Earth scientists to make testable predictions. And I would say that actually exceed the testable predictions, the ability of the evolutionary model. And it exceeds it, and this is, I think, really the kicker, on the most important arena of genetics. What is the mechanism of evolution? It is the mutation rate. It's mutations over time. That's the ultimate explanation. And it is the young Earth model that is taking the lead in this field, the most fundamental aspect, the mechanism of the evolutionary model. So Dr. Venema, again, has a fantastic intro in his first chapter, hypothesis that is not rejected. After many predictions and tests, becomes a broad explanatory framework, makes accurate predictions about the real world, a theory. He would say evolution fits that definition. I would say young Earth creation fits that definition. It remains a productive explanatory framework. Hopefully that's what you've seen this hour. Not only has it been failed to be rejected over 50 years, it remains a very productive and fruitful explanatory framework. We're working on the research questions right now. I think it's extremely exciting. Anyway, you can see my excitement. The question for you, of course, is, bigger picture. This is one small drop in a very bigger debate. Who do you trust? That's going to be the question. And whoever you trust, how do you explain the other side? So, hopefully what you've seen explains for you then why I'm titling my book Replacing Darwin, because I think we've gone beyond rebutting Darwin. That was the defensive strategy of decades ago. Now we've advanced so quickly, I think we can actually replace Darwin, and roles have now been reversed. That's what I'd argue, and will be arguing in this book. You can get the book chapter in chapter 10 of Searching for Adam is available on our table. We actually have 4 books out there, normally retail for $72. You can get them for $20, so it's a real saver. Again, we didn't have time to discuss the bigger general question, how do you get tens of thousands of species from just a few on the ark in a few thousand years? That's what I cover in this lay article series and much more in depth in my technical papers. So, thank you so much for the invitation, for the opportunity to present to you, and for staying on this late evening.
This has been, this conference has exceeded my hopes and expectations. I have heard both men speak in other venues, and I knew that they were both very bright, very articulate, but both of you even surpassed what I had hoped and expected. It was quite remarkable, your presentations. As a non-specialist, I'm always blown away by what you have to say, but not just that, just the way that you are able to take some really difficult concepts and are able to communicate those as well as you have. I want to apologize to the audience who have been watching over Facebook, the streaming video across Facebook. I think we've had over 10,000 different viewers this afternoon, so we've had a spectacular audience. The difficulty is that they've not been able to see the animation in the PowerPoint, which has put them at a tremendous disadvantage. The good news is that when we put the material on the CFC website, we will combine the respective PowerPoint presentations with the audio and video talk, and at that time, one will be able to see it. Of course, that'll be too late to help the students who are writing the reports because it'll take them a month to do that. So you all, unfortunately, are out of luck. You'll have to write it as it is. Dr. Venema, you just heard Dr. Jeanson and make his presentation. Let me give you the first opportunity to give your observations of what you've heard and perhaps questions you might have for Dr. Jeanson. And then after he does so, then I'll ask you the same question.
Okay, great. Am I on then?
Yes, you are.
Now I'm on. Good. First off, I want to say thank you to Nathaniel for your presentations. Very clear and articulate, and I appreciate them. Excuse me. I also want to underscore the fact that I think it's okay for brothers in Christ to have these differences and have these dialogues. So I hope that this can be a model for how to talk graciously about these issues. I recognize that for some communities it's seen as more of a central faith issue than for other communities, but I think even, like Answers in Genesis says, that this is not a salvation issue, right? This is an issue, one's interpretation of the early chapters of Genesis does not
You'll have to ask him.
Okay, we'll let that go. I think I've read that on the website.
Is that question one?
Just right off the top, I want to read something in my defense from the book that talk about where I say, there aren't young Earth creationists that are correctly trained in population genetics. And that's actually something that doesn't come from me. That actually comes from within the young Earth creationist community. So we've mentioned Todd Wood before. This is what Todd Wood has to say about it. So I'm basically just taking what he said and sort of rephrasing it. So Todd says this: the population reconstructions are complex and not easily understood by lay people right now. So creationist responses lag behind the current science, and the best your typical creationist can do is cast aspersion on the science. Until we have a creationist well-trained in modern theoretical population genetics, I think we will continue to have only unsatisfactory answers to these ancestral population reconstructions. So I guess maybe one comment to say right off the bat is that this is not just a conversation between young Earth creationists and evolutionary creationists. It's also a conversation within the scholarly young Earth creationist community. And one of the things that I've enjoyed over the years, I actually have a friendship with Todd. I haven't yet met him in person, but we email back and forth. To be completely honest, Todd says, Dennis's advocation of evolution makes me very uncomfortable. And that's reasonable. But he says, but we have a lot of common. And he says, we have Jesus in common. And Jesus, he says, is a lot to have in common. And I just love that particular turn of phrase. So yes, Jesus is a lot to have in common, absolutely. So that was just on that particular point. I guess what I'll be looking for in the coming years is, and this is, Todd is somebody who has read Carter, he has read Tompkins and whatnot. So
That's actually incorrect. So it's not actually a conversation within the young Earth literature, and I don't mean to be belligerent. He's a unique character. Jeff actually worked with him for many years, I think, at Clemson. And he has said, it's a real shame. Todd's really talented. He's reached out to Todd Wood. I've reached out to him to say, review my papers, and has refused. To me, conversation is a two-way street. You talk, you listen. I'll read his papers. I'll cite his papers. Jeff has reached out to him. Can you please review this? And he refuses to engage any of this. And so, to me, his statement saying, there's no young Earth creationists, I'm like, he also doesn't seem to be aware of Rob Carter's work. And so I know from my own personal interaction, he has declined to engage what we've published and shuts himself off. And he said on his blog, he says, what I really wish, my burden for the young Earth creationist movement is to have an independent voice, sort of an independent critic. And that's kind of, you read his blog, and that's kind of how he styles himself as. He says he's too tied to the young Earth creation movement to be that, but actually he's distanced himself very significantly. That's not something we go around parading, because we're not here to bash people, but that is the behind-the-scenes reality that we haven't made public just because we're not interested in bashing. But sadly, he has not shown any interest in conversing about it, but seems to feel no restraint about posting publicly things that are not true. So he's not actually having a conversation. He's one of these splinter groups that has their own following. You have to ask anytime someone says young Earth creationist, well, what's their means of accountability? You'll see things like on our website, other websites, arguments we think young Earth creationists shouldn't use, and you'll find out the young Earth creationists are quite offended because that's their favorite argument, and even creationist organizations, well, how dare you say that? And people come alongside, and I have to consider this data. They don't want to be accountable, and so, and we seek accountability. Like I said, at Ancestral Genesis, we have an editorial review board that reviews everything that goes out. We seek outside review, independent review. We'll have sometimes bi-yearly meetings on subjects. We had one on the Ice Age because there might be different views, and one exactly within the young Earth framework that works. We try to make it a continually self-correcting process, whereas Todd seems to have removed himself from that, not just that process and accountability, but doesn't even want to even talk about it, which is really sad because he's a very talented individual. But as an example, he published an abstract. So not only has he not done it behind the scenes, he's not done it publicly. To me, the best way for accountability is written peer review. You've probably experienced this as well. I could tell you other stories about how I've experienced it. People are resistant. And that peer review comes from the nature of science we discussed, as you laid out in the first chapter. Science is an inductive method of reasoning, You never prove anything in science, you can only disprove. We're all fallen human beings. There are things I may have missed, there are things Dr. Venema may have missed, and you seek other scientists to say, you know this methodology, what hypothesis have I not excluded to jump to my own? And in hopes that the group think will then arrive at the conclusion. So that's what we seek to do. And anyway, so a lot of what Todd has published, let's say on human chimpanzee genetic similarity, he published something in 2006. Most of Jeff Tompkins' work postdates that. Todd has published a few abstracts, which are maybe a paragraph, which, if you've ever read the scientific literature, that's not something you can critique. It doesn't give you methodology that you can repeat. It doesn't give you a way to critique it. So he really hasn't published anything, either on the topic, that we can evaluate. Where Jeff's methods are transparent. He's got detailed lists. So unfortunately, he takes the label, but again, it's not a copyrighted label, and he resists accountability on that front, and even resists our invitations. I mean, he sets himself up as critical, and I ask, my boss, hey, can Todd review these papers? And each time, no thanks. Just takes a very negative attitude, which is a shame.
Well, obviously I don't know the ins and outs of young Earth creationist sort of behind the scenes sorts of things. What I find interesting about Todd is that he's trained, he's a geneticist and has good training. And in my interactions with him, I've found him to be approachable and willing to dialogue. And yeah, I enjoy very much hearing what he has to say. So yeah. Let's see, what else should I ask you about? I'll fully admit to not being completely up to date on young Earth creationist baraminology. What does Answers in Genesis hold as the human baramin at this point? And I know Todd is an outlier on this point as well, because he includes an Australopithecine within the human baramin. What's Answers in Genesis' take on that right now? How far does the baramin extend?
And just a quick clarification, baramin is sort of the colloquial term.
Created kind. Yeah.
Baramin, yeah. Not colloquial, the technical term. That's why it's colloquial, if we understood it all.
Yeah.
So
my latest understanding is that Neanderthals are human, Denisovans are human. I'm just not sure where Answers in Genesis is at on that front.
And so there's a history to this field of baraminology. That's been sort of the term in the young Earth literature. I tend not to use it as much just because it tends to be a very specific meaning. But the basic idea is something I referred to earlier, which is we're not species fixity proponents. Again, you read scripture from our perspective. God creates kinds, not species. That's not the term used. And on top of that, the land, everything kinds are wiped out, restart in the Middle East. So it's kind of beyond me why the creationists of 1859 would somehow think that geography was fixed when you have a geography that's totally rewritten from a young Earth perspective about 4,500 years ago. So we would say there's limited common ancestry. Scripture, again, doesn't give you a whole list of terms. I'll give the derivation very briefly here, and again, you can read more of it in our literature. So Noah is commanded to take two of every land, air-breathing kind, male and female, for the purpose of propagating the seed or the offspring. So that then implies, the purpose is reproduction. Kinds must be able to reproduce. And so as a crude first approximation of what might a kind be then, we've done breeding tests. And since this is dependent upon a specific scientific test, where that kind boundary lies is going to change with time with the data that we have. So 50 years ago, you might not have very many breeding tests, and they might say maybe it's genus. And then, we can actually connect almost every cat species in a reproductive continuum. Now we're going to bump it up to the level of family. As we do more breeding tests, might that change too? And really, to me, the ultimate answer is going to, again, be genetics. Fossils aren't inherited, geography is not inherited, DNA is inherited. So that's going to be the record of ancestry. And it's going to go back to these questions of, it's going to go back to the standard of science. We're going to determine what the kind boundary is by seeing which explanations make the best testable predictions. If I say it's the genus and I can't predict anything in terms of genetic diversity, that's probably not a good hypothesis. If I say it's family and it's starting to explain what we see, that's the process we'd go about doing it. So then the challenge is, back to your question, what do you do with fossils? So I didn't mention the Neanderthal DNA when I was talking about 6,000 years for a variety of technical reasons. I think the question is still open. Most of my evolutionary colleagues would say that they think the DNA is reliable. They would all acknowledge that it's degrades from the ends. You're talking about stuff that has been sitting in the ground from a young Earth perspective for thousands of years, from an evolutionary perspective for millions of years. When I was doing DNA analysis as an undergraduate, we would stick stuff in the freezer, minus 20 degrees Celsius for a year. DNA would start doing weird things after a year, and we just toss it. So I'm highly skeptical, just based on that experience, that we're ever going to be able to recover some sort of reliable DNA from Neanderthals. And I've made technical arguments in my paper saying it doesn't even fit the structure. If anything, if you're going to say it's reliable from a young Earth perspective, you have to say it's some sort of hypermutatable. I think, again, the most likely explanation and most boring explanation is that it's degraded. So to me, we lack the tools. Todd has been trying to do a method where he tries to quantize the number of non-genetic features among various creatures or species, and seeing if you can find groups. Now, what's the challenge with that? Number one, it's not genetic data. Number two, it's highly dependent on which features you pick. And that's been some of the discussion in the young Earth literature, some of the responses to his work. Well, you're only looking at teeth and this. And actually, I asked Dave Menton, who taught anatomy and physiology at Washington University for like 33 years. So what do you think of Todd's method? He's like, well, it's exactly what I expect from a geneticist trying to do anatomical comparisons, basically, which is, that's not how you do it. So I think the jury is still open, I guess is a long way of saying that, because I think we lack the good tools by which to analyze it. I think it would probably be fairly unanimous that Australopithecines would not, going with what the criteria Dave Menton would use, saying here are the distinguishing features of the species we see today, chimpanzees versus humans. And in many cases, the fossils that are prominent and being debated may not even have fossil data for that particular distinguishing feature. Certain, let's say you're talking the skull, a certain bone up here in the nose, or you look at the fingers, or anyway, Dave Menton can speak that a lot better than I can, but that's where it's at the moment, that Todd has been using largely his particular methodology, which is statistical quantitative, highly dependent on which data you're using, and you can see that in some of the history of results, and not necessarily weighing distinguishing anatomical features more heavily than things that don't really distinguish species.
Okay, good. Okay, another question. One thing as a biologist that I'm often curious about with the young Earth literature is that many baramins, or accepted groups of relatedness, have DNA diversity within them that are actually far greater than human and chimpanzee differences. Mm-hmm. So I'm curious why that is the case, or how that fits in a young Earth creationist model. If we can see diversity in related groups that exceeds the human-chimp difference, and from a young Earth perspective, that's happening in a very short period of time, then why, how is it that we can baraminologically reliably locate humans as not related to other species?
So the question basically is, if you look at nuclear DNA sequences, there's a whole bunch of ways you can compare it. There's different categories of differences. Single nucleotide polymorphisms would be single rung differences, basically, where I have an A and you have a T. Indels, insertions and deletions would be, I've got a row of these 3 rungs and you're just missing those 3. You've got whole chunks. It might be, I've got this section of 3 million rungs or 500,000 rungs and you're missing it altogether. These are the sorts of things you'll see. We've got 4 to 4.3 million or so, 3 to 4 million single rung differences around the globe, and then a variable number of these other types of differences. Actually, the indels are more like, the small ones are 600,000 or so. All that background to say, let's compare species at the single letter differences. It's easier. And we have data for that, and it's data that I remember offhand, so it's even better. So for humans versus chimpanzees, the single letter differences in the regions you can align, again, ignoring that part that doesn't align, you'd have around, well, you saw it, about 26 million or so, 1% difference. Let's say you compare the tiger to a house cat, which we would say are part of the same kind. There are almost 50 million single letter differences. As a general rule of thumb, we'd say that species within a family have a common ancestor. So the great apes belong to a single family. Evolutionists would put humans in the same family. We wouldn't, but chimpanzee, bonobo, the 2 orangutan species, 2 gorilla species, all part of the same kind, we'd say, came from a common ancestor on board the ark. The orangutan, gorilla, orangutan chimpanzee differences, I think, anyway, they are 3.4% different. I think it comes out to around 100 million differences. So 10 times, well, maybe 5 times higher than what you see in humans. So what does that imply about ancestry? And so this goes back to the question then of what are the tools we're using to determine it? And are there patterns? Are there numbers of DNA differences? And none of those criteria seem to be good tests. Ultimately, the test is what testable prediction does a model make. So right now, I would say it would seem that God created, let me back up. The data I didn't show you, some of which is in my article series and my technical papers, is young Earth creationists like myself. I've argued that this idea of pre-existing diversity, God creating Adam and Eve with differences within themselves, applies universally among species. That the cat ancestor on board the ark was front-loaded with a whole bunch of different DNA differences, and those then have led to the origin of species. Actually, the primary cause, we would say, the origin of species. So from that perspective, I would say it would appear that God created animals with more potential for diversity than humans. So as it relates to the question of baronology, for many years, the criterion has simply been breeding. And then now, because we're getting DNA data, we can finally bring genetics into it. And so we're now having to develop criterion. What sort of tests can we use to identify common ancestry? Well, what testable predictions does a model make? So if I can say, let's use mitochondrial DNA. Let me think of actually a better example. Bovids. So Bovidae family, 135 species alive today. Everything from cattle to sheep to goats to African antelopes. Very diverse family. Cows and sheep differ by 10% of their DNA, I think. So 10 times higher than human chimpanzees. We'd say maybe they have a common ancestor. They're clean animals, so it might get a little sticky, but that's a side note. Mitochondrial DNA, that's where I was going with this. So there's maybe 2,000 differences between cattle and sheep at the mitochondrial DNA level. Humans versus chimpanzee, there's only 1,400. Humans versus humans, you saw the data, 80 to 40. So how do you explain the fact that you have cattle and sheep potentially sharing a common ancestor, but there are thousands of differences apart? Humans, we'd say, have a common ancestor, and not related to anything else, less than 100. That'll probably be a function of DNA differences, mutation rates. So no one's measured the mutation rate in these bovid species. We can make testable predictions, and really that's what it's going to come down to. So I would say we've made retrodictions for humans. We can make testable predictions for Khoisan peoples, other people groups in which it is yet to be measured. I can make predictions then for sheep and cattle, and the success of this model will be seen on how well those predictions bear out in reality.
As a lay person listening to you, I just want to make sure if I understood what you're saying. Are you saying that on the ark there was a common ancestor for sheep and for cattle, and that sheep,
potentially?
Okay, you are. What's potentially mean? Just so I'll understand. Are you saying then?
For sake of argument, yes, but because they're clean, and then there's the question of 7 of the clean, 2 of the unclean. Could it have been that there was 3 different looking representatives? I don't know. So for unclean, cats would be an easier example. Two cats, and you have everything from tigers to house cats to jaguars in between.
For my simple mind, sheep and cattle are pretty dramatically different species. Mm-hmm. So whenever I hear someone saying that's a common that they had a common ancestor, well, I got in trouble for everything I thought about that. I thought that was quite a remarkable thing. So I just wanted to make sure I understood that. Do you have any other questions you want to ask him before I turn him loose on you?
Sure, sounds good. Just maybe 1 or 2 more, if that's
okay.
Then it'll be my turn, right? Then I'll be on the hot seat.
Yeah.
Just a quick question. When you say those examples of pseudogenes with mutations and nested hierarchies, do young Earth creationists accept that within accepted baramins? Like, would you look at gorillas and chimpanzees and orangutans and say, yeah, they would have inherited that mutation from a common ancestor?
I would like to.
Okay.
So it goes back to the question of how do you identify common ancestry? And if there was a mutational signature, I would embrace it quickly. But I would say what we call a mutation and what we don't is virtually impossible at present because of the non-intuitiveness of DNA. So again, back to the language analogy, we speak less than 1% of the DNA language, as far as I'm concerned. You just have to look at the history of genetics. Back in the 40s, 1 gene, 1 protein, 1 function. I'll talk to people from a generation ago. I taught a class at Southern Evangelical Seminary on biology and origins, and we had a gentleman there who's like, everything I'm learning is the opposite, or this is totally different from what I learned in school. It's changing that fast. And so what you might look at a textbook a decade ago is now being totally overturned. And so things that look like mutations, because they look like broken genes, I think of pseudogenes as a specific example. So when I was in graduate school, one of the newfangled, unexpected ways of DNA function is actually something near and dear to my heart. So what I worked on as an undergraduate is RNA. So DNA is transcribed RNA translated to protein. It's the intermediate between the two. We're working on RNA regulation. My boss was always, RNA is the underappreciated, this is the big deal, and we need to make more of a stink about it. Well, he's basically been vindicated, because the amount of literature on RNA function is more than I can tally. I mean, there's reviews written on this. Anyway, back to graduate school. So one of the shocking discoveries, I think made originally in plants, was RNAi, RNA interference, small, short RNA snippets that bind to, and I think there's base pair differences there, they look like mistakes, bind to other RNAs and regulate the function. And I remember sitting in class and one of my profs saying, Andrew Fire was in Massachusetts. He was one of the guys, he's going to get the Nobel Prize, and sure enough, got the Nobel Prize for this. So what are pseudogenes? They're sometimes antisense versions. To me, what comes to mind immediately is they're going to function in some sort of RNA regulation. It's just too close. Does it look like a broken gene? Yes, but to me that's decades ago biology, and I'm not being insulting. My point is, I don't disagree that it looks like a broken gene, but I also see this massive literature that's saying there are all these unexpected things RNA do, and then I look at the fact that we've actually functionally tested less than 1% of the genome. To me, the genetics community has done a really bad job making predictions in terms of function. And that's not an intelligence thing, it's just because we're dealing with a foreign language, a highly compressed, it's beyond the best computer code, is really what it comes down to. I mean, we write books, we don't write books that can be read forwards and backwards. DNA can. You've got proteins. There's a phenomenon, and I've written part of this in a paper, one of the first papers I wrote, protein moonlighting. So one of the 1940s ideas was one gene, one protein, one function. So some of the mitochondrial DNA proteins. Well, we know the function. It functions this particular enzyme complex. It takes this chemical, transforms it to this, releases water, whatever, transforms oxygen. This particular enzyme takes glucose and it removes this group, and it's at this step. This is classic. It's known. Well, now we're discovering serendipitous things like, this enzyme that functions in sugar metabolism is now binding to DNA in the nucleus. Anyway, moonlighting. Proteins are doing things that you don't even anticipate. Let me explain it by analogy. Proteins, if you think of our bodies as biological construction projects, we start as a single cell, and all of this is built in the womb, basically, so that the human form we recognize as this is apparent by birth. Proteins are one of the main tools for building the body. Protein moonlighting is analogous to having a multifunctional tool, but what human engineer builds a handle that is a hammer, saw, drill, and a whole bunch, 10 other things in one hand. I mean, we don't even design, it's science fiction what's going on at the cellular level. So
if I'm understanding you correctly, if I could just jump in. You're saying that the DNA code works as if I were reading a book. If I read it this way, it would be a poem, a Shakespearean sonnet. If I read it the other direction, it would be perhaps a newspaper clipping from the Washington Post, and it works both ways.
And so again, some of the classic paradigms, you can go to some of the textbooks I have, they'll say, even in those things we thought we understood, let's say this particular DNA sequence codes, is transcribed to this RNA, which is translated to this protein. There's this talk of synonymous codons. So in short, there's 20 amino acids, 4 different DNA letters. There's different languages in your cell, and 4 different DNA letters can't code for, one letter then can't code for all 20. It's groups of 3. 3 DNA letters code for a single unit of a protein. And I was taught the third, so there's some redundancy. That's the point I'm trying to get at. There's some redundancy in DNA coding for protein. Well, I was taught, there's this wobble position 3. If you look at textbooks, statistically, then, it can be variable. Well, now we're finding that actually it plays a functional role. And there's even reviews being written about this now, that these aren't just, it tolerates error. The cell uses different redundant elements to slow and speed the process. I mean, the level of information compression into every single letter is really, it boggles the mind. So that was a different example than you're asking about, but it's a crazy field.
One more question?
One more question.
Okay.
And he's gonna
Although
ask you.
there's some things I would say in response.
Please do. We can have a dialogue.
No, this is a dialogue.
Oh, okay. Well, I don't want to take up too much time, but there are cases where we see pseudogenes, where we actually see this, many cases actually, where we see the same gene in a functional state, say in mouse, relative to the human genome. And we have the ability to determine its function experimentally in mouse. So one of the examples I gave was along those lines. So what do you think of those kind of pseudogenes, like where we actually have experimental evidence that it has a certain role in mouse, it's in the same little block of genes in the human genome, yet it's non-functional, or at least it can't have the same function that's in the mouse. So why is it that we see that pattern? I'm curious, from a young Earth perspective, what does one do with that?
I would say, and you can tell me if I'm articulating the argument correctly, there's an element of homology in the argument, and there's an element of non-function. You have the homology in that it's shared, but then the other element of the argument in human looks like it's non-functional.
Sure.
So, to the homology element, I go back to design and saying, human engineers reuse common patterns for similar purposes. What would be the purposes then in DNA? And that's to me where it gets sticky, because we're so bad at determining purpose. I mean, the whole idea of protein moonlighting, the whole idea that RNA is doing all these crazy things, and is it weekly, monthly, that some new discovery is coming out? So, can I predict at the moment what that human gene is doing? No, but in 5 years, I have a pretty good guess that I will, because it's going that fast. And it's not what we anticipate it to be. And there's so many interactions in the cell. So one of the hottest fields right now is the 3D arrangement of DNA in the nucleus. That the cell not only uses DNA according to sequences that exist before and after the gene, but it's how it's compressed and uncompressed and wound together. There are so many serendipitous discovery, I think it'd be foolish to try to assign a function. I don't know that anyone could assign a function. And what it turns out to be will probably blow our minds. And I think it would be highly premature to say it's non-functional because of the trajectory. So many paradigms have been overturned in the last decade. I mean, I graduated 2003 from undergrad, and I have my undergrad textbook. And so, what is that, 15 years or so, and things are being overturned then. It's so fast. Like I said, I would love to have a tool to say, this shows the signature of ancestry, this one doesn't. And I don't think I can do that, given how fast things have changed in terms of what we say is functional and what we say is not.
One last question. I asked this question of Georgia Purdom back in 2011, I think it was. She and I had a similar dialogue at LeTourneau University, and it's out there on YouTube if you want to see it. I asked her this question, and I'm curious, so I remember her response, and I'm curious of your response as well.
See if we're consistent.
See if you're consistent or not. Not that I'm trying to catch you out or anything.
I'll keep my phone in my pocket in case she's watching and going to text me.
Good,
we can't cheat.
Yeah. And I may not get it exactly the same, but I asked her something to the effect of, do you think, is there any evidence that could potentially convince you that your position on origins is incorrect? Is there any, can you conceive of evidence that might convince you that you've taken the wrong tack on things? I'm just genuinely curious.
It's a good question. And I would say it's more of a philosophical question.
Yeah, in some ways, sure.
There's a scientific element, a theological element, and a philosophical element, and I'll answer each of those in turn. So, the scientific element, I've given falsifiable predictions, and I'm happy to go test in the lab. Let's go do it. Let's go test this. What have I been saying all along, though? Take evolution versus creation as an example. The non-evolution, the non-genetic evidences. I've said, this fails to reject evolution, fails to reject young Earth. There's all sorts of hypotheses that it probably fails to reject. Within the young Earth model, we'd say there's, Scripture clearly teaches these things, and on these things it's silent. And even in the arenas where it's silent, there's a whole lot of hypotheses you can create. So, let's say I make a prediction on the mutation rate and it's false. Does that mean it's only this or it's only this young Earth position or not, there's probably 5 other young Earth positions that fit it as well, that it could create. So basically what I'm getting at is science is it's simultaneously a very poor and very powerful tool to know the world. It's very powerful because we're sitting here with our iPhones and technology is advanced, and that's due to science. But it's very poor, especially when it comes to origins, because the further back you go in time, the less and less you know about what potential competing explanations exist. And so, in that sense, science is very weak. And I think it would be foolish to base such a gigantic question such as origins and the truth of Scripture on such a weak and unstable discipline. Powerful discipline, but inherently weak in terms of absolute truth, and such big, big questions that touch on so many topics. So religiously, we have a very specific hermeneutic that we adhere to, so it would require hermeneutical change to change my view then on Scripture. That's tied then to philosophical as well. So what is my view of origins? Origins is a multidisciplinary question in which science plays a role, a limited role, because all it can do is eliminate competing explanations, and there's a whole lot of them that need to be eliminated. And science is inherently pragmatic. At the end of the day, there are some, and I would like to consider myself one of them, who are interested in knowledge for knowledge's sake. But a lot of technology is driven by, does it work? Can it work? Great, move on. We want to send men to the moon, so we're going to do the physics until we get it right. Do we need to do the physics until we understand the last detail about the nature of matter? No. We've got men to the moon. It's going to get funded. I mean, there's a money roll in all this too. So that also makes science a weak way to answer big questions. It's a great way to know the natural world, but when we're dealing with natural, supernatural, and all that, you can see how this becomes weak. I love science, and I love to do science, and I think it's super exciting, the research I'm doing, but I also recognize the inherent limitations and the need to bring in Scripture and other things as well. So I fully embrace making testable predictions, recognizing that there's a whole lot more. And you being an old Earth creationist, I was fully anticipating you, the wheels turning and saying, well, how does this work with old Earth creation? I'd say, well, there's probably testable predictions I could come up with there too. Just as an example, a practical example. Does what I show you tonight distinguish or eliminate old Earth creation? Fuzz Ron and you, Ross, would have to answer that. And it might not. There might be some things I've shown you tonight that might seem to reject evolution, but not reject old Earth creation. You see where I'm going with this. The point is, I haven't considered everything that's out there. And probably neither Dr. Venema or I will ever consider everything that's out there. That's why science changes so much as our knowledge of the world grows. So scientifically, do I have hypotheses that can be falsified and changed? Sure. Will that change my young Earth view? It's going to be pretty tough because there's so many things that are still compatible with a young Earth view, other hypotheses on specific topics. And to me, that's true in general of science. I mean, even hopefully, as you've seen tonight, when it comes to potential evolutionary explanations for the different compartments, you have inherited differences, you have selection you can deal with the time scale. I think you'd agree. Even within the evolutionary framework, it's hard to find an either or. It's either this element or this, and this, versus either this, and this, and can we find one where there's what I call in the book a category 1 experiment, where it's going to, if it goes this way, it's clearly young Earth, if it's this way, it's evolution. Oftentimes, most of the time, it's never the case. It kind of favors this way, but over this way. So I think that's the reality of what we're dealing with. And so scientifically, falsifiable predictions, yes. Religiously, it's a hermeneutical question, and that's entrenched in philosophy as well. So.
Well, I am glad that you pointed out that I'm actually in a mediating position. And that's one of the reasons I have enjoyed, one of the many reasons I've enjoyed this night is that I'm actually in a position literally in between the two of you. And so, so that has put me in the proverbial catbird seat in more ways than one. Dr. Jeanson, excellent presentations, excellent answers. Now it's your opportunity to make observations and ask questions to Dr. Venema.
So first of all, thank you as well. And hopefully you've seen, I've really enjoyed, especially that first chapter and the way nature of science laid out. And even as you're presenting the language change and such, sitting there thinking about speciation in 4,500 years. And that's a useful analogy. And here's how we see DNA change. And actually, if you read, anyway, I'm supposed to ask you questions, so I'll stop and ask you questions. So I presented some strong challenges on mutation rates and such. What would your response be to those, or some of the things I put more punchy?
Sure. Certainly on the variation that we see in mitochondrial DNA. Mitochondrial DNA is very constrained, right, because it's very small, and there's very little, as you know, as a biologist, there's very little space in between genes on mitochondria. And in fact, many of the genes actually overlap one another. So I wouldn't expect, and I don't suppose you would either, just as a biologist, that the differences, the raw differences that we would see between different groups would solely be the result of raw mutation without any selection that's actually removing deleterious mutations from the population. So I wouldn't expect to see those raw differences merely just be like a sort of a neutral clock of certain mutations. I was actually curious about your thoughts on Neanderthal DNA for that specific reason, because if you include Neanderthal mitochondrial DNA, then of course that makes the problem. There's a lot more variation there to account for. So I would see definitely selection for that particular limited, because of the sort of dense nature of the information that you see in a mitochondrian's genome. When it comes to nuclear DNA, I would be more comfortable with thinking about it in more neutral terms, because the evidence we have is that a large amount of our DNA doesn't have a sequence-specific function. So I'm more comfortable as sort of a neutral, without considering the effects of selection more generally for nuclear DNA for that reason, which is not to say that there isn't strong natural selection on certain parts of nuclear DNA, absolutely. So that would be my general thoughts on that particular question.
So I'll put it more punchy.
Sure.
Because I did it in general to an audience. I'll put it to you. So what testable predictions would you be able to make from that mindset? Can you predict the rate of mitochondrial mutation in Khoisan? Can you predict the rate of mutations in cats?
Sure. It really depends on having an understanding of the functional constraints on that particular one. So again, it's a lot easier to do that kind of analysis when you're looking at sequences that are almost neutral. And one of the things that we can see in nuclear DNA is you can actually look at nucleotide substitution rates when you compare related organisms. And we see that nucleotide substitution rates are actually much larger or much faster in regions that don't correlate with things that we think are functional. So if you look at gene coding sequences, they have a substitution rate that's far below what we would see in, say, introns, the bits in between coding sequences that are spliced out. Now, there's still some functional constraint there as well, of course. We see even faster substitution rates in pseudogene sequences, for example. So these are lines of evidence that give us confidence that there are different parts of the nuclear genome that are under different selection constraints.
So I'm going to be naughty.
Pardon me?
I'm going to be naughty.
Okay.
He's gonna be naughty.
All right. The gloves come off.
What number could you put on it, and is it something you'd be willing to test in the lab?
For mitochondrial rates?
For any species.
For any species.
So I'm claiming I can make a prediction
Yeah.
for cats.
Sure.
Can you?
Or for the Khoisan, for example. I'm actually
yes.
very fascinated to see you make that prediction, and I'm interested to see if that's because that's actually that takes guts, right, to put it on the line like that.
Are you
right?
all daring each other?
The trick is
there going to be a double dog dare somewhere?
The trick is that if you're taking functional constraint into as part of the equation and not just sort of raw mutation frequency, then you actually have to have a good understanding of the selective constraints in those different sections of that genome and also in the environment that they're in. And that's something that we don't have ready access to for a lot of different species. So it'd be tricky from an evolutionary perspective to put a specific prediction on that.
So I'm going to be even naughtier. So if you can't make a prediction for it, should it be considered science?
Well, it's not that a prediction couldn't be made. It just requires a large amount of information that I'm not sure that we have ready access to. So it certainly could be done if enough effort was put to it. Absolutely. And we have seen good predictions made, say like with the incomplete lineage sorting. We predicted the percentages in advance before we looked at the gorilla genome, before we looked at the orangutan genome. And that was because we had data we had the relevant pieces of the puzzle that we could use to make that prediction. So we certainly could make that prediction for mitochondrial DNA, but it would require resources that we don't have at ready hand. But they could be accumulated if enough effort was put into it.
So if I understand the independent lineage sorting correctly, the prediction was after DNA variation was already obtained for these species.
No, it was prior to sequencing their genomes.
Yes, I guess I'm saying we already had genes, other sequences.
We had a
Subsections of the
small
genomes.
subset of it, yes.
Yeah.
And we had estimates of population sizes that were part of that. But we had not looked at a full genome sequence. And we were actually making the prediction off of a very small subset of the DNA. Yeah.
I guess my point is, I thought in my mind, being naughty, it seems a little bit like cheating. Can you predict whether an average guy off the street can you predict how far he's going to hit a baseball? And you let him hit 10 times, you get the average, and you say, yeah. How much of a prediction is that? Prediction is, I predict 450 feet, and I know nothing about you. That to me is a prediction, I think. That's what I'm arguing I can do for mitochondrial DNA without getting all these. So would you say in that light, young Earth creation should be treated as science?
Science doesn't matter where it gets its hypotheses from. That's one of the beautiful things about it. You can have you can have a wide variety of where hypotheses come from. And scientists are actually very pragmatic people. They don't mind what your worldview is. They don't mind what your views on God are. One of the things I experienced, and you probably may have as well, as a PhD and postdoc in secular settings, is that what counts in science is predictive power. What counts in science is whether or not your ideas bear experimental fruit over a long period of time. So nobody really minds where the predictions come from as long as they fit with the data that we have and that they make good predictions going forward.
So would you say the young Earth view should be considered science?
It does science, like I said, it doesn't matter to me where predictions come from. That whole demarcation question of what science versus non-science, philosophers get lots of, that's how they get their paychecks on science versus non-science. So yeah, what matters in science is its ability over a long period of time to make accurate predictions.
Any other questions before we go to the audience? Okay. Well, we do have some questions, and so let me just go back and forth. Dr. Jeanson, let me ask you this first one. A watcher has asked this. It said, would the young Earth position expect genetic evidence regarding the fall? Would there not be an expectation of a massive recreation after the fall? In other words, would not young Earth creationism require a massive genetic rewrite given what young Earth creationism thinks happened to animals, that they were transformed from plant eaters to carnivorous? If so, if the answer is yes, what research is being done or exists to support this claim? That there would be a massive alteration of bodily morphology and complete system rebalance. Does that question make sense?
And it's a common question. I'll restate it this way. So young Earthers would say no death before the fall, including animal death. No carnivory, put it that way. No pathogenic E. coli, at least no disease, that sort of thing. So where does this come from today? What's the origin of these particular species and structures? Where did lions get the ability to tear flesh? Where does pathogenic E. coli come from? Where do viruses come from? And there's 4 major explanations, none of which, so the answer is basically no, I'd say, massive genetic rewiring. Here's why. You can get pathology simply by changing the environment. E. coli in the gut is good, E. coli in the blood is bad, potentially lethal. So you can find, forgetting the name of the bacteria, there's soil bacteria that do fine in our body bad. So changing simply the balance of the environment, the harmony of the environment, internal environment, external environment can lead to pathology. Degeneration. You've got pathogens that are dependent on us as hosts or other creatures as hosts. If they once were free living, that may not have been a pathology. So you've got change in environment. You've got degeneration as an explanation for pathology. You've got repurposing. I might be wrong in this story, but I want to think the history of the Nobel Prize. Did Nobel invent TNT? I want to think he thought he invented something hoping this would end all wars, and then he saw what his technology was being used for, felt terrible, but so funded the Nobel Peace Prize. Maybe I've got this story totally backwards. Anyway, the point is, you can use TNT for good, you can use it for destructive purposes. You can use sharp teeth to tear flesh, you can use it to open coconuts and watermelons. Not that there are necessarily those plants around back then, it may have been different. The point being, you can get aggressive behavior simply by repurposing it, whether it's good or bad, it depends on how it's used. So that might be a simple explanation. Some might say, well, there's a whole digestive system, short for carnivores, long for herbivores. Pandas have a short digestive system, and they subsist on bamboo. So it's not quite the relationship that we necessarily predict it to be, and we're looking at one point in time. And then last is, maybe more related to the question, latent genetic information. Here's how I'm going to derive it. There are some plants, there's different types of photosynthesis in different types of environments, whole biochemistry, whether you're in the desert or the tropics. And you might think, from a young Earth perspective, God must have created these separate, totally different biochemistry. Well, it turns out some of these plants can interbreed. So you start dialing the clock back and saying, okay, and sometimes the difference between the two is just a simple genetic switch. So, going back to this idea of front-loading genetic information. Now this will get into Calvinist theology. So, let's step on some toes. There is the potential, depending on your theology, that God could have front-loaded information that could have been switched on after the fall. And that's all I'm going to say about that theological topic.
Can I be naughty for a minute?
Yes, go ahead, be naughty.
I kid a little bit, but we see exquisite adaptation for carnivory in the fossil record in present-day animals. I would have a hard time looking at an 8-year-old boy and saying T-Rex ate coconuts. I really would. I'm just curious what you think of that. I'm sorry, that's a little off the cuff, but yeah.
No, and that's a legitimate question, and you could think of it, here's maybe my first response. You've got lions who seem designed to kill, and we'd say are relatives of house cats. So how much of, and I'd say most of those differences from a genetic perspective were front-loaded, and it's through a process I didn't have time to get into, the result of post-flood speciation. So the first question in my mind is, where in the speciation timeline do they arise? And who are their relatives? Yes, there's things that look designed to kill. And so a design argument would say, God made them that way to kill. And that's a strong argument. And that then gets into the theological question. And I won't give away my theology, but I'm comfortable with God knowing what's happening and front-loading it in and seemingly making them designed to kill. So I'm fine with saying no coconuts. Yes, they look designed to kill. And we've lived in a sinful world from a young Earth view for most of our history. So we're looking at judgment. And God has exercised judgment repeatedly throughout history. On a global scale, we'd say, from a young Earth perspective. So it's a harsh reminder of what we'd say goes back to the Adam and Eve question. How's that for a tie-in?
So are you comfortable with the idea that the elephant walking through the Garden of Eden maybe stepped on some insects and killed them?
That gets to a fuzzy issue of where do we draw the line of life versus non-life? What's the nefesh, basically?
And I'm glad to, I'm glad to have that theological discussion with you.
Or should I say it's a squishy line if it's
you go.
stepping on.
Squish away. All right, let me. Since we're talking theological questions, Dr. Venema, one that you have gotten a lot, several have asked. You've done a very powerful. You've done a very good job of making a very powerful argument for that change is on a gradient, that is gradual, and this is true of the hominids. And so when we ask that population of 10,000 that were, what was their previous generation, and they're just imperceptible in the changes. Then where does human uniqueness in the Imago Dei fit into this?
Great questions, absolutely. And biologically, it's a very difficult answer. But biology isn't the whole story, right? So even among Christians who accept evolutionary creation, there's a range of views on these topics. Some hold to some sort of punctate event where God calls our species into relationship with him. And typically they would view the Imago Dei as a relationship, that it would not be predicated solely in our biology, but it would be this relationship that we have. So we're called to be stewards of creation. We're called to subdue and rule, as we see in Genesis. So image in that case is then not so much connected to our biology, although, I mean, it's going to have to take a certain amount of biology for us to have a relationship with our Creator and fulfill that relationship and that stewardly responsibility that we have. One of the reasons why I'm a bit uncomfortable in connecting image to particular biological characteristics is that we can look at human variation in the present day, and any characteristic you happen to pick, you can find individuals in the present day who are outside of that or on the edge of that variation. So we can say, well, okay, intellect is the part of the image of God. As a species, yes, we need a certain amount of cognitive capability to relate to our Creator. But then we have individuals who have severe developmental disabilities, and they are as much made in the image of God as anyone else. So I prefer to look at image of God as a relational aspect. And among evolutionary creationists, some are comfortable with that relationship happening gradually over time, or coming into being gradually over time. And other evolutionary creationists feel that there would have been a sort of a punctate event where our species was deliberately and punctately called into relationship with God. So there's sort of that range of diversity there.
Okay.
Yeah, good question though.
Any comment or question on that? Okay. Then back to Dr. Jeanson then. You talked about it's not a cloning event, that Adam and Eve were not a cloning event. However, Eve was taken from the rib of Adam. So, one observer asked, couldn't that or shouldn't that be understood as a cloning event?
How much time do we have left?
Just a few minutes.
Because there's the obvious question that no one's asked yet, but I don't want to miss it. I'll answer that one quick.
Okay, go ahead.
Yes, she's taken from his side, so that would imply then if Adam was created with two different versions of his DNA, she would have been virtually identical to his two different versions, except for sex chromosomes. Obviously, she's female. And what's interesting is, if you look around the globe, most of our DNA varieties come in one of two versions, fitting that sort of idea. So, is she a clone of Adam? Yes and no. There's all sorts of differences within Adam, differences within her. So you could call it that, but usually when we talk about cloning, we're saying making a genetically identical version. Is she genetically identical? Well, if you want to call a whole bunch of differences identical, I guess so. So there's that element, but it seems to fit what we see in biology.
All right. Any question or comment?
So I'm just curious on that point. So you're saying that there would be new created differences in the process of making Eve? I'm just trying to understand it from a...
No, in that sense it might seem to be like cloning. She's made like Adam. She has, except for the XY, obviously, because she's female.
Adam has an X, so all you need is two copies of that X, right?
Well, I guess I'm saying not completely identical because she's got to have something that makes her female.
Sure.
But otherwise, would his chromosome 22, his two different pairs, look the same as Eve's? Potentially.
Okay.
But both of, Adam's number one of that pair and number two of that pair look different from each other, as do Eve's from one another. And then that'll lead to all sorts of, the potential for variety is incredible.
Okay. Interesting. I'm just curious. I'm curious about that just from a technical point of view, because some young Earth creationist materials seem to posit 4 different starting points, but what seems like you're saying is that there's just really 2. So I'm curious. I'm kind of just...
A priori, you might expect that. Rob Carter's one who pointed out to me saying, you do have that from the rib. Does that mean it has to be the same? Maybe, maybe not. And then he said, well, most of our DNA differences come in 2 versions, implying then it goes back to just 2 different versions rather than 4.
Interesting.
We're running out of time, so let me ask the question that you started with, that you asked to Dr. Jeanson, what would cause you to do a rethink? What type of evidence or what kind of new development might make you say, okay, in other words, what kind of discovery or hypothesis might Dr. Jeanson present that you'd say, I'm going to have to really think about this?
Interesting. Well, as a scientist, scientists are always open to new information. It's actually interesting, Nathaniel, your answer was very, very long and detailed. Your colleague's answer a few years ago was not. It was very brief, and the answer was no evidence, right, because it's a scriptural issue. What I see on this issue, so yeah, scientifically, if young Earth creationism, over the next decade wins the day scientifically, absolutely, scientists go with the evidence, no problem. I don't see that happening. There's lots of lines of evidence that we haven't talked about tonight, geological evidence, those kinds of things that I think make the young Earth creationist position scientifically untenable. But say, it's conceivable that, scientists are always open to new evidence, so it's conceivable scientifically that it could shift, I suppose. But I would be extremely surprised. I would also be surprised if we, could I conceivably shift my views on heliocentrism? You bet. It could well be that there's a better model out there. Now, that better model is going to have to incorporate the data that we already know about, and it's going to have to do a better job of explaining it than the mainstream data, or the mainstream models of explaining it now. But any scientist would always put a caveat on anything. Even things that are extremely well established in science can be shifted, but I don't think we're going to change heliocentrism anytime soon, and I don't think we're going to change evolutionary biology anytime soon either.